Everything. Okay, perfect. And he's very active in providing talks and seminars at all levels, universities, high schools. So I think, yeah, so he's traveling most of the time with this very, very important activity of outreach. So thank you, Luis.
Thank you, Vicente, for this very kind introduction. If you can stop sharing your screen, I will be sharing mine.
See. Okay, now.
All right, thank you. So I think everyone is here. All the students are here. So let's get started. And today we have a busy morning, and Vicente was inviting me to share with you two topics that are most relevant, but they are often neglected in the education of a PhD. And this is why I understand Vicente wanted to have this opportunity for Episeedlink MSCA Action students, as you all are. So we will split this morning in two different topics which are related. First, I will navigate with you through the different parts of ethics in research, which is important, which is fundamental, which is mandatory. And so you all have to know this, and you all need to comply with all these different regulations. And this will take us about a couple of hours, from now to 11:30. Then we will pause for about half an hour. And at 12 at noon, we will resume. And I will devote the next 2 hours until 2 in the early afternoon, discussing on research integrity, which is the other pillar that you need to be aware. So let's get started with ethics in research. So ethics is a topic that often generates fear, because people think that ethics is the same as moral, is the same as philosophy, is the same as sometimes religion, is the same as normative and law. So how do we make this the understanding, the true understanding of ethics? So this is why I always like to start my classes with some very basic definitions. So what is moral? What is the morality? Well, you should know that there is not a single morality. There is not a single moral. Moral is an agreement that a given society decides and upon what is good and what is bad, what is right and what is wrong. So this is setting codes of conduct that are adopted and they eventually govern the human behavior. And those contravening and then breaching these codes, they are the ones that are collected in the laws. So basically, you are acting against the law when you are doing something that for that particular society is considered to be bad, is considered to be wrong. This is what we call the moral. And as you can understand, we might have a European moral, or you might have a Spanish moral, a French moral, a German model, because not in all countries the values are perceived the same way. Not to mention, if we go to Asia or we go to other countries in South America or in other parts of the world, so what is considered to be good or bad might be relatively different in all these countries. So when do we come across ethics? Well, ethics is a discipline that is included in philosophy. And as is here written, ethics is a critical reflection about morality. And ethics is providing us a way to sort out and to fix some dilemmas that might be appearing when we try to decide whether two different behaviors that each one of them independently can be considered to be good, But eventually you have to decide which one of them is the one you will be favoring. So I guess the best way of understanding this is with an example. I guess we all will agree that animal welfare is a value per se. Nobody wants to have any harm or produce any harm to any animal. This is a value. This is a good behavior. This is considered to be right, to be a good thing. At the same time, you can use animals to develop new therapies for treating devastating diseases. And of course, this is also a very good thing to do. So does it mean that for every single use of animals for developing therapies, these animals will be or should be approved? Not at all. This should be done case by case. And for each one of these situations, you need to decide whether it will be justified or it will not be justified, the use of animals for developing a new therapy. So here we have these two different situations that are both good, but the ethics help us to decide which one of them we will be following according to the details of that particular project. So this is ethics in general, and when the ethical issues are applied to healthcare, to biological and to medical sciences, then we're talking about bioethics, of course. I guess everyone understands that ethics is appearing when you are aiming to collect biological fluid from a human being. So here we have collecting of blood. Of course, this blood is part of this person. This blood identifies this person. It contains cells, and the cells contain DNA, and the DNA identifies that person. So we should handle these samples with great care and respecting the dignity and the confidentiality of this. So I have the impression that we all agree that we see an ethics issue when we are collecting blood. We also see an ethics issue when we're using animals in research. So it is clear that we cannot do whatever we want with animals. There is a very defined law. There is a very confined and restricted law that prevents the use of animals in research if there are alternative methods available, and only allows the use of animals if it is truly justified and if there are no alternative methods. And I guess these are the two aspects that probably are more distant from what you're doing in Episeedlink, because these are not related with plant molecular biology, but you will see that these are topics that clearly people will understand that they have an ethics dilemma that needs to be solved. But I want to tell you that what you're doing, I'm assuming that you will be, for instance, investigating the diversity of species in this forest. This is a research project probably closer to the type of research that you're doing. There is ethics behind this project as well. You cannot just go to the forest and you start delimiting areas, and maybe you have to take samples, and maybe you have to make a hole in one of these trees in order to estimate its age and to estimate its health status. And you probably will be collecting plants and individuals here and there. You cannot do this without first argumenting why are you doing this type of experiment and what is the potential benefit that this community or this forest, this mountain, this village may have from your project, and what would be the risk associated. So eventually you need also a permission. You need a permission in order to do this experiment. It is not as simple as I go to the forest and I start doing research. Nobody cares because nobody sees me and I'm here collecting samples. Absolutely not. You are in nature, you have to respect the nature, and you have to get the appropriate and the mandatory permission before you start doing this experiment. So this is also ethics. And this is ethics as well as it was ethics to collect blood or to use animals. We have also ethics when we go to this Caribbean beach. We are in this Caribbean beach and we are lying on the sand, and the sand is not properly made of sand grains, but there are the skeletons of many invertebrates that are there, and we think that they are very funny. We think that they are very funny, and these are very tiny beasts, very tiny animals. And perhaps we have a tube, and perhaps we take one of these animals and we put these animals in a tube, and because we are researchers, when we arrive to the lab, we make an extract from this very tiny beast, and out of the extract, we obtain a substance, and this substance we're giving them to cancer cells in culture, and all of a sudden, the cancer cells stop dividing. We have found a cure for cancer. Great. Well, not so great, because we have been using a biological resource, a genetic resource from a country that is not ours. And we have to respect their rights. We have to respect that they need to benefit in the corresponding share about the results of the research that we're doing. So this is the same as to say, we need to respect the Nagoya Protocol on access to genetic resources and the fair and equitable sharing of benefits arising from their utilization, which means that whenever we go to a given country and we are collecting a given individual, a plant species, because we think it's nice, because we want to import it illegally to our country, well, this is not only an illegal operation, but it also contravening this Nagoya Protocol, because any potential result, any potential use of these plants and their derivatives in research will have to respect the rights that this original country has regarding all their genetic resources. So this is something that often is forgotten, often is neglected, and whenever you are coming across the use of some plant species or some biological samples from other countries, you need to remember that first of all, you need to obtain the permission. You have to respect this Nagoya Protocol and obtain the permission. Then we have a number of ethics situations that I wish to discuss with you, because these are very important, especially in this scenario in the European arena. So the first thing I want to discuss with you is the dual use. You probably know that there is a number of research projects that they can have a civil benefit, but they can also have a military benefit. Well, there is nothing wrong in principle with the military use of research, but the only thing we need to know, and this is very important, is that the funds, the money, this support that we are obtaining from Europe for civil research can never be used for military purposes. And this is because the military research have their own pot, have their own money. So this is done and handled differently. We cannot use civil money. We cannot use these euros from MSCCA Action or from ERC project or from Horizon Europe project. And we cannot use this technology for military use. And sometimes people say, well, I'm not using this for military purposes. Well, you might not be using this, but the technology that you are developing eventually could be used with such a purpose. And then, since you are getting public money that is to be used only for civil research, then you need to make sure and you need to put all the different blocking so that nothing from your research gets used by the military world. So let's give a couple of examples. The Horizon EU research project, that is to say, the European money, the European Commission money, has an exclusive focus on civil applications. and activities intended to be used in military applications or aiming to serve military purposes cannot be funded. And in a case in which such a dual research might be contemplated, then you have to apply a regulatory framework. There is a given regulation that has to be respected. And these are the regulations. You don't have to worry. I will be sharing with you these slides. I will be sending these slides to Vicente, and you will get all these different regulations. So this is not the point now to go global law, but at least you need to know the existence of these laws and the emphasis they have in these different aspects of ethics. For instance, here there is a table in which you have to decide whether what you're doing can be interpreted to have a military use, or it is, as it should be, exclusively used for civil applications. I give you an example from my type of research, which is genetically modification of animals. Could be the same in other species, but this happens to be in animals. This is atrin. Atrin is an antithrombin recombinant, and this is a recombinant protein that is produced in the milk of the mammary gland of these goats. So these are transgenic goats, and this was one of the first animals to be approved by EMA, the European Medicament Agency, and later on withdrawn years later because it was found out that it was not that useful. But anyway, the point here is that here we're using technology for a civil application. We are producing a protein that it is an anti-clotting agent, and this to be used for those people that they have a mutation in the corresponding gene and they are not producing enough of this protein. Normally, this protein is obtained from plasma with the corresponding risk of infections of viruses that can be found in plasma. And one solution to get away from these infectious agents is to use this recombinant protein produced in the mammary gland of these goats. And I think we all agree that this is a beneficial application. This is on paper a civil application. But the very same application, the very same technology can be applied by another totally different company, and this is Pharmatin. Pharmatin is creating Protexia, and Protexia is an antidote for anti-nerve agents. And I'm referring to sarin gas and other organophosphorus agents. So what are the mode of action of this Protexia? You probably remember from biology classes that neurons are communicated through neurotransmitters. And one of the neurotransmitters is acetylcholine. Acetylcholine is released from the emitting neuron and is received by the receiving neuron. And this is how we transmit the neuronal impulse. But immediately thereafter, the acetylcholine is reaching the destiny neuron. There is an enzyme which is called acetylcholine esterase that will break down acetylcholine. And this will be interrupting the communication. And this is what you need to allow in order to have different levels of communication. So you activate the next neuron, but once it's activated, you don't need to reactivate it or to maintain this activation. What happens with sarin gas and other organophosphorus agents is that they compete with the neurotransmitter for the site of action or for the place where this esterase is holding this neurotransmitter. Therefore, the acetylcholine cannot be broken, and this results in a hyperstimulation. And this hyperstimulation generates, when this is affecting a number of neurons, a total collapse of the body, and then the person dies. And this is why these nerve agents are so dangerous. And this is why they are used as war weapons, as biological weapons. So Protexia, it is a recombinant protein creating it in the lab that is having the same holes, the same places to to bind these organophosphorus agents. Therefore, it's used to clean up the extracellular media from this sarin gas and allow the esterase to break down the neurotransmitter. So in other words, this is an antidote, and this is to be used in biowars, in wars in which biological weapons are to be used. And this is clearly a military application. It will not be surprising to you if I tell you that Pharmatin has been funded by the US Army. And clearly, this is an application that needs to be funded. If someone thinks this is interesting, this needs to be funded separately. So this cannot be funded through civil funds. So I cannot use European funds from that are to be focused on civil applications to develop these antidotes. If someone is interested, then you need to find out the corresponding military funds. The same is true if we change fields and we go into engineering. And this is the constellation of satellites of Galileo. You know that Galileo is a long-standing project in the European Union. And these are meant to be replacing the GPS satellites in order to precisely locate objects and people and vehicles in the position. So let's imagine you are developing batteries for these satellites. And this is, of course, a great thing to do. This is a civil application. But you need to be aware that the same batteries can be used for other satellites that are probably used to locate and to decide where a rocket will be launched and where a rocket will be exploiting. And this is not science fiction. This is what is happening every day in Ukraine, for instance. So the very same technology, which is developing better batteries, can also have this dual use. And then we need to prevent the use from the military fields, and those applications that are corresponding for military applications, they need to be funded separately, not through our civil funds. And last but not least, batteries in cars for electric cars. This is obvious. They can have a civil use, and as we see again in nowadays Ukrainian war, this Russian invasion, so we can have these military vehicles which are also propelled by electricity. So this dual use is more often present than everybody understands or appreciates. So you need always to reflect the type of research you are conducting can have this dual implication. If you have this dual implication, you need to decide what you're going to be doing. So how you will be approaching this. There is a subcategory of dual use, which is misuse. And here we are contravening research integrity, and we probably contravening some laws, because it's not only that we are using the research funds for things that we shouldn't be doing, but also that these experiments that we're doing, they can represent a very important risk. They might be dangerous to the population. They might be dangerous to human beings. They might be dangerous to the environment, to nature, etc. So as before, best is to use an example. When I was using this example before the current pandemic, people thought that this was a bit of extreme. But now we're just escaping, or we're just finishing the COVID-19 pandemic caused by this coronavirus. And now we know what we're talking about. So you know that a bit more than a century ago, there was this influenza pandemic that was wrongly called the Spanish flu. And I'm saying wrongly called because that had nothing to do with Spain. So this influenza pandemic had to do with the end of the First World War. The soldiers, they got infected, and when they returned to their own countries, they were spreading this influenza pandemic in the world. So this was affecting about 500 million people, and 10% of them, 50 million at least, they were dying out of this pandemic. So this is a serious virus. This was a particular serotype, H1N1, that was appearing at the time. And there have been researchers that decided that this is a biological entity that needs to be investigated, because this is a very powerful viral particle. It was highly contagious. It was highly powerful for replicating and etc. And there was a biologist that requested permission to do this type of experiments. So they would go to a cemetery and they will dig and they will obtain the carcasses or the cadavers from people that died a century ago. And from these remainings, they will try to obtain a virus. Of course, they can do this, and of course, they might be a justified project, but this needs to be done under very restrict biosafety measures. Because if you're using such a viral agent, which is so powerful, you need to prevent their spreading, this accidental spreading into the population. And this is exactly what this virologist, the message that he was receiving. He can only use this if he is using these viral particles in a biosafety laboratory type 4, level 4, which is the highest. And because everyone can understand, if you have such a virus sample and you're spreading this virus in the street, you will be creating a disaster. And of course, this is something that is considered to be a misuse. You're doing a pure biological and a pure research project, but the consequences of working with this project might be affecting the other human beings or might be affecting the rest of the planet. Another example for misusing. Drones are used very often in research projects, and the examples I will be providing are coming from my expertise, my experience in the ethics panel at the European Research Council in Brussels. So for instance, drones can be used to inspect some mountains and some hills that are where you're growing some trees, maybe olive trees, and you want to know which one of these olive trees has been infected by this bacteria, which is called Shillelagh fastidiosa. And this is already a name that is already alluding to the dangers of this bacteria. This bacteria is to be transmitted by insects between different plants. And once the bacteria reaches one of these trees, there's not much you can do other than removing the tree and avoiding the tree to be spreading out this bacteria into other individuals. How do you find out which tree is being infected? Well, there are circumstances in which you can use an infrared camera, and this infrared camera might be adjusted to determine the levels of chlorophyll. And the levels of chlorophyll can be detected, and those trees that will be affected, they will have a lower level of chlorophyll, and then you will be able to detect and to pinpoint all these different trees that can be removed. But of course, when you're flying these drones, you're not only flying over trees, you're also flying over people. And of course, you might be collecting images of people that for a given reason are there. They are just having a walk in the fields, or this is because the olive trees are next to a village, and then all of a sudden you are coming and you're flying over some person, and this person doesn't need to explain why he or she is there, why in that precise moment in time is there. So we need to respect this confidentiality. And we have two options. Either we stop flying, we go down, we land, and then we try to collect the informed consent by each one of them so that they allow us to use their images in our video, which is probably nonsense and not very practical. Or what we do is exactly as what Google Street is doing in Google Maps. So when they are collecting these images to help us finding a building, they are blurring the faces of the people so that people cannot be recognized. So either way, this is to prevent the misuse of your research activities. Because each one of these persons that you see here now in the street, they have all their rights not to be detected, not to be highlighted. And so this is, I guess, these are examples where you see that dual use and misuse can be appearing in many of your different research projects. So please think about this. So ethics means safety, security, privacy, respect, well-being, welfare, environment. And we have to care because no research project can commence without an ethics approval. And this is to ensure that we are fully complying with norms and regulation regarding our due respect to humans, animals, plants, and environment. We are obliged to maximize benefits and minimize risk and harms while considering the needs that are associated with each research project. And this is not for us to discuss. So they are not asking us for an opinion. The ethics approvals are mandatory. You cannot start a project without the corresponding ethics approval, without reflecting whether your project will have or not ethics uses. And most projects have ethics uses, especially most projects that are often not recognized by the PIs. And so they need to be amended, they need to be corrected, because you need to discuss all these ethics uses and how you will be dealing with them. You also need to remember that if you have an ethics approval, this is protecting you. This is protecting your research and the research staff that are involved in this project. For instance, if you are given an interview to a journalist and this person is asking you about your research and you are using genetically modified organisms, as is very often the case in the plant world, then you can be sure that you can very openly and transparently talk about your research because you have the permission, because you have already discussed the ethics issues of your project that includes the use of genetically modified organisms. You got the clearance for this project, and then you can talk about this. So nobody will tell you that you are doing something that is against the law because you are strictly following the law. So as a matter of fact, getting this approval is protecting you, and basically you're showing to the rest of the society that you are adhering to the accepted ethics standards. There is a number of existing rules and laws that we need to comply regarding human subjects, regarding animals, regarding plants and environment, regarding GMOs, regarding the use of biological agencies and databases and etc. And I'll be going through all these different topics. I understand that maybe for you is not so much interesting the use of human subjects or the use of animals, because this is probably not the main purpose of your research, but I think it is important that at least for once you hear about these different ethics issues, because all of a sudden you might be producing an antibody in a transgenic plant. This is possible today. You might be producing a plasma protein recombinant in the leaves of Nicotiana. And this is an example that is possible. And the resulting product, you might wish to test it in a clinical trial. And all of a sudden, even if you are a plant molecular biologist, you ended up involved in a clinical trial with human subjects, and you need to know what are the rules and what are these scenarios that you need to respect. So the bioethics, it's a rather young discipline. And actually it appears, not surprisingly, after the end of the Second World War. And as you see, there was the Nuremberg Code in 1947, the Human Rights Declaration, the creation of the United Nations, the Helsinki Declaration, the Belmont Report, and in all, what we have is a number of principles that they need to be respected. And these are the basic principles in bioethics. And this is important that at least you hear about these 4 principles at least once in your research life, in your professional life, because these are principles that are fundamental and are present in all the rest of legislations, either explicitly or implicitly. The first of these principles is the principle of non-maleficence. We should not do harm. We should not create troubles when we're doing research, for instance, on human beings, but this can also be applied to plants, to the environment, or to animals. We need to find an equilibrium between the potential benefits and the associated risk that are involved in our research project. So basically, we translate this not doing any harm by safety. So when we are analyzing a proposal for a drug or for a therapy, before we know whether this therapy will be useful or not, what we need to demonstrate that this therapy is safe, is not creating any more harm than the one that we want to cure. So principle of non-maleficence is the first always. So first is not to do harm. Second is to do good. Is our duty. We have to do something that should be beneficial. Once we know that a given treatment is safe, it must be efficient. You must evaluate the efficacy. So it is clear that it will not be producing any harm, but it is also clear that it should be producing some efficient, some good consequences on the subjects or on the environment. And when we're talking about the human subjects, doing good is not an arbitrary good that we decide as PIs and we impose to the human subjects. We need to accept that we first need to collect the informed consent, because that links with the third principle, which is we have to respect the autonomy of the people. So we cannot recruit and involve human subjects in a clinical trial without their informed consent. They need to be informed what are the risks they will be facing, and they are to decide with total freedom whether they will be participated or not. So this is why when we are testing for a COVID-19 vaccine, we're not talking about subjects, we're talking about volunteers. So human subjects are volunteers always. So they need to decide by themselves whether they will be participating or not. And fourth and last but not least is probably the one that is often more neglected. And this is the principle of justice. And this is the one that guarantees the equitative access to these potential benefits to everyone that can benefit. If we are producing a therapy to treat a given disease, this therapy should be available to everyone having this disease, irrespective on the economic capacity of these people. So even the poorest people, they have the right to access to these therapies. Well, as you have seen in the World Health Organization, WHO, was releasing the COVAX alternative, and the COVAX alternative was sort of involving the richest countries to purchase enough COVID-19 vaccines for third countries in the world, in South America, in Southern Asia, and in Africa mainly. Well, this proposal did not work as expected. And as you probably remember, not all countries purchased these different vaccines. And this is why, while in Spain or in other European countries, we are over 90% of coverage of people vaccinated, there's still some countries in Africa where the coverage is below 10%. And this is because they didn't have this access. So this principle of justice, which should be applied in all cases, normally is the one that is suffering the most. So how do we translate these basic principles in bioethics? Well, The principle of non-maleficence, we translate it into safety, evaluating the safety. The beneficence, we translate it into efficacy. The respect for autonomy, the voluntarity, and the principle of justice, the universality. So, and these are the 4 principles that will be appearing one time after the other in all the different legislations. When you are approaching a project to be funded by European Commission, such as Horizon Europe or such as this Mary Sloboska-Curie action, these are the ethics issues you need to take into account. So we will be going one by one. So at least that you have some headlines, you have some knowledge about all these different uses. To begin, we will begin with human embryonic stem cells and human embryos. So basically what we're applying here is the Oviedo Convention. The Oviedo Convention that was signed by not all countries, France and Spain and Italy, for instance, they signed this convention, but Germany did not sign, or United Kingdom did not sign, or China, or Korea, or Japan, or the US, they did not sign for different reasons. In the case of UK, because they thought it was too restrictive, and in the case of Germany, because they thought it was too weak. They wanted a stronger legislation. Well, there's a number of articles, but regarding human embryos, there are 2 articles that are important to remember. Article 13 basically says that we are not supposed to be modifying the genome of our descendants. So we cannot germline treat our embryos. We cannot impose a genetic modification in embryos that eventually will be resulting in a human being. This is forbidden. And this is what is applying here in Spain. And in Spain, we have 2 articles in the law. If these articles are contravened, if someone is crossing these articles, then it will be sent to jail. It will have to pay a strong fee, and it will be inhabilitated for working in research for a number of years. So this is serious stuff. But of course, there are countries that they are not obliged because they are not signing this Oviedo Convention. And Article 18 is also important because Article 18 says we can use embryos, human embryos for research coming from in vitro fertilization, for instance, redundant embryos. Once a couple have used their embryos and they have obtained the children they wanted, there are some remaining embryos. These embryos, they can be donated to another couple, or they can be also donated to research. And then basically these embryos will be used and they will be destroyed. They cannot be maintained, they cannot be implanted. But what Article 18 says is that we cannot we cannot make human embryos only for research purpose. We cannot take sperm and eggs. We cannot do in vitro fertilization and the resulting embryo to be used for research. We can only use those embryos that have been reconstituted for reproductive purposes, and they will be eventually donated by the corresponding couple. So this is illustrated by this genome editing example in human embryos. This is one of the first uses of human embryos donated from redundant in vitro fertilization embryos. This is in UK, and this is Katinia Khan from the Francis Crick Institute. That was published in 2017, and she was evaluating the role of a gene comparing human and mouse embryos. Most of what we know from mammalian embryos comes from rodent embryos, and you might think that what is true for mouse embryos, it should be true for human embryos. Well, not always, and this is why you need to do some experiments with human embryos. For instance, if you inactivate this OCT4 gene in mouse, the blastocyst, which is the last stage in the embryo pre-implantation development before it implants in the uterus, the blastocyst is fairly okay. Whereas if you inactivate the same gene in the human embryos, this is totally distorted and not even a blastocyst is being produced. So blastocysts are compromised, And this is a clear example that the same gene is producing different phenotypes in different species. There is no way you can anticipate how a gene will behave in a given species if you don't do the experiment in that species. And this is why this is the type of experiments we can do. But these are experiments done with embryos, human embryos that have been donated for research purposes by the corresponding couple and have been approved by the corresponding committee. So what we have in Europe is that it is allowed to use human embryonic stem cells. So these are cells that have been obtained from the inner cell mass from inside these pre-implantation embryos. These embryos have been destroyed and you have been obtaining these pluripotent cells and you have established these human embryonic stem cell lines. If they have been produced before and have been pre-registered, in Europe, you have the allowance to do this. They need to be registered in the EU registry. But what you cannot do, and this is because there's some countries such as Poland, Germany, Malta, and other countries, they are not allowing the creation of new human embryonic stem cell lines. So they are not allowing the destruction of human embryos, of novel human embryos for creating these lines. For instance, this is not true in Spain. In Spain, we can create, we are allowed to create these lines, but in other countries, such as, for instance, in Germany, you cannot do this. And this is something you need to respect. And because you know that these decisions are taken with the adoption of all the member states. We have 27 member states at the moment. And because there's some of these member states that they are not accepting this, so this type of research will not be funded by the European Commission. Another case is the induced pluripotent stem cells. These are somatic cells that with the transfection of some reprogramming factors, they have been converted into pluripotent and they can be derived and resulting in organoids, for instance. These are mini brains. So these are a collection of different cells that they can represent a very simplified version of an organ. So these are alternative methods that they can be obtained from these iPS cells. So this is regarding the human embryonic stem cells. So what about involving of humans? So human subjects in research. Here, we're talking about volunteers from social or from human sciences as well. We need to take into account whether these persons that we will be recruiting, they can give the informed consent, because maybe they are in coma and they cannot give, and then the relatives, they will have to give on their behalf. Maybe they are vulnerable, they are suffering from a very terminal cancer disease. And of course, these people, their decision when they are asked whether they would like to test a therapy that can be very dangerous, it is they are vulnerable and their response might be conditioned to their situation. Or they might be minors, they might be children, and of course children, they cannot consent, so their tutors, their parents are the ones to be consenting, and etc. So basically, the highest protection always goes to newborns, to babies, even when you're doing non-invasive type of experiments. This is a non-invasive functional magnetic resonance, the fMRI, and if any one of you has been explored by this machine, you probably will agree with me that this is not very comfortable. It might be non-invasive, but you need to stay stiff for 20 minutes or 30 minutes. You should not be moving. And if you are an adult, this is okay. But if you are a 2-month-old baby, there is no way that this baby will stay without movement. So you have to fix the head. And if you fix the head, and then it becomes already an invasive procedure. And it needs to be very well justified before this is to be approved. Always counterbalancing the benefits versus risk. What we're protecting is the patient, the human subject. What we're regulating is the use of the samples. And the samples have a very wide meaning. is not only the biological, but also the data, the genetic data. And we have to ensure respect for people and for the human dignity. The informed consent, which is what you are giving in a paper, in a piece of paper, when you go to hospital for a small surgery, they might be using the piece of tissue that they will be removing from your body for research purposes, and they will be providing you a paper, and they will say, please sign here. You have the right to read everything. You have the right to ask for what purpose they will be using this tissue, and you have to right to find out what they will do if they find something that you did not expect. Let's say they are investigating your genome for a given rare disease, but all of a sudden they realize that you are carrying a mutation that will be resulting in a neurodegenerative disease later in your life. Then you must be informed about this possibility, and you have the right to know, and you also have the right not to know. And the right not to know, it's very important. And this is the right that applies in this family. This is a representation of a family. The square is the father, and the circle is the mother. And this couple, they have 3 daughters. The second daughter is affected by a given disease, which means that probably father and mother, they are carrying this mutation. And they might wish to know whether the other first and third daughter, they are carrying as well the mutation. They might think it is interesting to know, but if they would know this, they will contravene the right not to know that assists these other 2 daughters. Because at the moment, while these daughters are still children, they are not reaching reproductive age, it has no health significance whether they are carriers or they are not carriers. The only importance to know whether they will be carriers is whenever they will be adults and they will decide to have children. Then they will have the right to know whether they are carrying the mutation or not. And they might decide to know it or not to know it. But if the parents, apparently innocently, they decide to know whether these other daughters are carrying the mutation, basically they are vulnerating, they are breaching the right not to know that is associated with these other people. That this is very important, and this is what we have to respect. Human cells or tissues. So sometimes we're not using a person per se, but we're using the corresponding cells that are coming from this and from this. We need to know one important thing, that is why human cells always constitute an ethics issue. Well, there are many human cell lines that are people using in the laboratory. And no matter which of these human cell lines you're using, you need to remember one thing very clearly. Each one of these human cell lines, some time ago, they were inside a human being. So in other words, each one of the human cell lines you're using correspond to a person. And because it corresponds to a person with a name, with a date of birth, with a country of origin, and etc., we need to pay the due respect and the due dignity. And so this is why working with human cells is not the same as working with mouse cells. People might say, well, there's no difference. They're both mammalian cells. Well, there are no ethics issues associated with the use of mouse cells, whereas there are ethics issues, very important ethics issues. You have to declare the origin of these human cells because they correspond to the piece of the body of a given person sometime in the past. You can obtain them commercially, you can generate it between your project, or you can obtain it from another research project, or eventually, which is the best origin, you can obtain it from a biobank. So this is the situation we have in Europe at the moment. The central path, which perhaps it was happening, and I wish this is not happening anymore at the moment. So many years ago, there were researchers, they had friends in hospitals, and when the given patient was subjected to a surgery, They will say to the pathologist or to the surgeon, they will say, why don't you take a piece of extra tissue and you give it to me, because I will investigate the presence of the protein that I'm doing my research on. And in case we find out that it is interesting, we might be requesting an ethics permission. But let's find out to begin with. Well, this is totally forbidden. This cannot be happening. There is no direct connection between the patient and the researcher. There's only two ways you can get into the human cells. First, and the most recommended, and this is why we have thicker arrows, is a biobank. Every hospital has a biobank. That's where they hold all the samples that are taken from patients. Patients, they're signing in, they're donating these samples to this biobank, and the biobank is anonymizing in the cases that it should be anonymized. It will be maintaining the confidentiality, and it will be distributing these samples to requesters, to researchers, that they need to justify what are they going to do with the samples. And in all cases, we should not forget that the property of these samples is always from the patient. So the patient might have donated a piece of their liver from a given biobank, and maybe 3 years later, he or she decided that they don't want to collaborate with research anymore. They don't have to provide any reasons. They don't have to argument. They just have to call the biobank and say, I am whatever name, and I was depositing a piece of my liver some years ago, but I'm no longer interested. Please destroy my liver. And this is exactly what's going to be happening. And those researchers that have received pieces of their liver will be contacted, and they will have to remove their livers from their freezers and destroy them. And if this is already being used in research and generated data, they will have to remove their data. And this is absolutely mandatory, period. There is no other way we can escape. The property of the samples always belong to the patient. And if the patient is collaborative, wants to collaborate in research, fair enough. But if the patient decides for whatever reason, they don't have to clarify why they stop trusting research. If they decide not to collaborate any longer, they have the right to request the removal of their pieces. Well, the biobank, as I said, this is the best way of accepting. You have Clarice, do you have a question?
Yes, Luis.
Okay.
Thank you.
Please. Because
so
I don't see you, yeah, the chat, I see only the video. So if you want to ask a question, just pop up with the camera. Okay.
Okay. Okay, so it's very fast. So I'm just wondering, what if you find you find out the cure to cancer or something like that with that patient's liver? And he doesn't want the research to be done anymore? Do you have to destroy all the data?
This is a very important question. And at the moment, according to law, you will have to destroy this sample, okay, even though you think that there is a larger benefit for the human mankind because a cure for cancer has been found out. So at the moment, there is some high-level discussions regarding the ethics of fulfilling this obligation. So the ethics of removing samples that have been already been positive or instrumental to develop a therapy. So this is something we haven't found out the solution. And this is something that at the moment, we just have to apply the law. And the law says that if the patient wants to remove their piece of tissue from research, he or she, they have the full right to do so. And there is nothing we can do to prevent this from happening. All right, we continue. So the next thing is collections. What are the difference between collections and biobanks? Collections is a group of tissues or biological liquids or samples you get from patients that they can only be used for a given disease. So I'm collecting samples for diabetes, and I'm using them only for diabetes. And my colleague, which is working next door in the lab next door, he is investigating multiple sclerosis. And he wants to use my cohort of patients because he thinks that there is an interference between multiple sclerosis and diabetes. Well, he cannot. He cannot use our samples because they are part of a collection. The only way you can use a sample for multiple purpose is if you deposit this sample in a biobank. There's a number of things that have happened in the past, and I wish to share it with you just to highlight the relevance of the bioethics of ethics in using human cells. You might know, or perhaps not, because most of you are plant molecular biologists, I understand. You know that one of the most popular cell lines in mammalian studies is HeLa. HeLa is a cancer cell line. It derives from cervical cancer. It was established in 1951, and it's been used in thousands of publications and thousands of patents. What is the point that is normally forgotten about HeLa? Well, HeLa stands for Henrietta Lacks. She is the person that was visiting her gynecologist because she had some troubles, and they find out that she was having a very aggressive cervical cancer. And as a matter of fact, she was dying out of this metastasis associated from the cervical cancer. These gynecologists, they were collecting some samples from the cervix. And when they were collecting samples and they put them into culture, for their surprise, they found out cells that they were living forever. They were dividing forever without any need. So basically, these cells only required culture medium, and they will be replicating endlessly. And this was one of the first cell lines that was established, and this was distributed. There was a company being formed. These samples were sold, and this is now present in nearly all research institutes in the world. What was the problem? The problem was that nobody asked permission to Henrietta to use this part of their body for research. Nobody. And how do we come across this lack of permission? Because in 2013, it was a German team that decided out of the blue to publish the genome of the HeLa cells. They thought, well, these are cells that are used by everyone, and we should be able to publish, and they will be helpful for everyone doing. Well, the granddaughters of Henrietta, they immediately complained because upon publicly releasing this genome, they were also releasing parts of their own genome, which they have all total right to be kept confidential. And there was a big controversy that was being created, and eventually the publication of only an edited part of the HeLa genome was released, just removing all parts that might be sensitive and that might contain information. I guess this is the best example to highlight why do we have to care when we're doing research with human cell lines. The best way of understanding bioethics is if you read this book, The Immortal Life of Henrietta Lacks by Rebecca Sloot. And if you don't want to read the book and you have Netflix, for instance, you can see the movie that is being made out of this case, The Immortal Life of Henrietta Lacks with Oprah Winfrey. This is a very good movie, by the way. And it's a movie about bioethics without talking about bioethics. So people who watch this movie, they realize about the power and the relevance of bioethics without attending a class on bioethics. So personal data. Personal data, what do we have to care regarding personal data? So what is a personal data? Well, personal data is your name, a picture of your face, your date of birth, your personal address. But personal data is also a tattoo you have in your arm. This is also personal data because this tattoo that you have in your arm is identifying yourself. A personal data is also maybe your signature, how you sign a given paper. These are all personal data that need to be respected. And especially you might have the permission to use some of this personal data, age, health status, for instance, for a given project. But later on, this data, you might think it's relevant for other purposes. But these other purposes, you didn't get the permission from the first subjects. So you have to go back to the people and you have to request this permission for this secondary use. And this is always forgotten. This is very often forgotten. So I have some data I collected 5 years ago from a number of people, and I want to use this data now for a different purposes for which I was collecting the data. Well, I cannot unless I get the corresponding permission. And this is very relevant. And if you don't have this permission for secondary use, your project might be scientifically fantastic, might be scientifically excellent, but you will not be funded. Well, we have this General Data Protection Regulation, which is normally it's called GDPR. Everybody realized about GDPR in 2018, in May. Probably you remember 5 years ago when you start getting messages from companies requesting you to confirm that you are willing to still receiving these spamming, these advertisements. Well, they were not just being kind, they were preventing from being fined, because this GDPR is the fundamental change is we're going from opt-out to opt-in. You're purchasing a television in a shop, and when you are creating an invoice, there is a small place where they say, click here if you don't want to receive any advertisement from us. This is in a very tiny letter. Normally people don't see this, they don't click there. And this is happening also, what's happening in the hotels. And if you don't click there, basically they have the right, or they have the right to spamming you. Now this is forbidden. Now the text should say, click here if you want to receive advertisements from us. Basically, you have to opt in. You don't have to opt out, but opt in. This is the fundamental change in the GDPR. So the breach has to be investigated, and this is empowering subjects. We have the right to be forgotten. For instance, an image is taken of any of you in a very peculiar or circumstances. Probably you are not very you're not very proud of that particular image, and this image apparently is distributed. Well, you have the right to request all servers to remove this image. You have the right to data portability. You can approach your hospital and you can go with an USB key and you can tell at the registration desk, please save into my USB key all the data, all the clinical results, all the X-rays, all the blood analysis that you have from me since I was born. Because these are data that belong to me. Before it was very difficult to get this. Now you can get this data and you can use this data and you can offer this data to a researcher that you're trusting because you think that he or she might be helping your disease. Or you can associate with a patient association and you can collect the data of all the different patients and you can contribute to research. So before, the researchers were seeking and were sort of trying to hunt for patients. It's now the opposite. The patients are hunting for researchers. The patients are looking for researchers to use their own data. This has to be started from the very beginning. This is called privacy by design. And basically in every institution, we have the DPO or the data protection officer, which is taking into account all these different aspects. This is also to be included in the informed consent. So they have the right to, as we discussed with the person who was asking this question. So this they have the right to be removed from a biobank if they decide to do so. There is some also ethics in social science. And the difference here is that for most countries, and this is the case in Spain, we don't have a precise law. We have a law that is regulating the use of biological samples, but we don't have a law that is regulating social research. What is social research? Well, you basically, someone is calling you and you are being interviewed on a given subject. So this is for many years, this was considered to be out of ethics. But of course, this has some ethics implication, and we have to respect all this dignity, integrity. We have to protect these vulnerables. We have to share the benefits. We have to minimize harm. We have to describe the groups that we will be approaching. We have to provide clear descriptions of the recruitment. And most importantly, we need to take into account these situations that are often used in the social science and humanities. Use of deception in research. Basically, you're cheating because you hope through this cheating, you hope to get the true answer from the person you are interviewing. Well, basically, you are manipulating your subject. And you have, in some cases, it might be justified, but this has to be very well argumented. And at the end of this exercise, you need to fully transparently explain what happened to the person you were interviewing, or cover research. You are asking for a series of questions and you want to derive a conclusion from some of these questions because you are not describing exactly what is the main purpose of your research, because if you describe, you fear that the answers will not be as free or they will not be as spontaneous as they should be. So this is very complicated and this needs to be taken into account. So the question is that for most countries, there is no legislation, but if you want to apply to European money, there is this October 2018 regulation in which basically it's a guidance with recommendations that you need to fulfill if you want to receive funds to do this type of research. I will be passing over this, including the use of internet research and social media, because basically this is something we all do all the time. When we are using an application, we immediately accept all the different claims and we don't read these claims. And because we want to use Google, we want to use Gmail, we want to use Google Maps, and we don't know, or we are not taking care about all the data Google is collecting from us, from our movements, from when we're visiting different shops, the types of movements that we're doing, and et cetera, et cetera. This is fundamental, and this is yet to be properly regulated. So we have some homework to be done here. Use of animals. Use of animals. I will be briefly touching on this. I understand you are not using animals, but at least you need to know that the use of animals is probably the most protected regulation that applies to research that we have in the European Union. What type of animals? They will be always non-human vertebrates, and that includes non-human primates. They might be genetically modified, they might be cloned, and they might be endangered species. Who do we want to protect? Of course, the animals, not the experimentator, not the PI. What do we have to regulate? The use of the animals in research and for teaching purposes, and what we intend to preserve, their animal welfare, minimizing harm, pain, or discomfort that might be caused by this research. We had principles for biomedical applications. We have also principles for animal research. And these are the famous 3R principles. 3R because they were established in 1959 by Russell and Birch. And these are referring to replacement, reduction, and refinement. And this is the basis of the European directive that applies in this case. Replacement means that we cannot use animals if there are alternative methods that avoid the use of these animals. So it's not a question of our opinion. It is mandatory to use the alternative method if this method is already existing. So we have to check and we have to review. There is a number of web pages. This is the ECBAM. This is the official web page for alternative methods in the European Commission. And we have other databases in which we can see the existence of alternative methods. And if there are, we have to use them. So definitely this is one way of complying with the first principle. This is already happening. This is what I did in the 90s. I was investigating regulatory elements of genes, doing hundreds of transgenic mice. And I was linking different parts of promoter and upstream sequences with a reporter gene, let's say LAGZ. And I was finding out where the regulatory were, because I was trying to reconstitute the expression pattern of a gene. Nowadays, I can avoid most of these animals, so I definitely don't have to use animals anymore, because I can compare the genomes of different evolutionary related mammalian species, and I can do comparative genomics, and where there will be a functionally relevant regulatory element, hopefully this element will be conserved throughout these different species, and it will be a best way to find out where these regulatory elements are, instead of doing experiments with animals. Replacement also has some moral attitude, and this is different depending in which country you are. Here, meaning in the Western countries, in the European Union, we care about pets. So we have a lot of respect for our pets, for dogs and for cats, less respect for pigs, even less respect for mice, less respect or less empathy for birds, for amphibia, and particularly we have the lower level of empathy, we have it with fish. Don't ask me why, but that's a fact. And so, believe it or not, the use of zebrafish, which is increasing in research, you know why? You know why it's increasing? Because zebrafish, believe it or not, are considered alternative methods. Of course, they are vertebrates. Of course, they are animals, but they are accepted as alternative methods, in this case, alternative to mammalian systems. So they are vertebrate as the mammalian, but they are non-mammalian, and they can be used without much of a problem. Of course, the highest empathy goes to our closest species, macaques, or any other non-human primate. We cannot use non-human primate for whatever thing. They are only to be used for reglementary use or regulatory use. For instance, you are developing a COVID-19 vaccine, and a vaccine needs to be tested, and this is mandatory, needs to be tested in a non-human primate after being tested in several rodents and perhaps in mice and hamsters. But before it can be entering a clinical trial, in particular phase 2, which is where you evaluate, you assess the efficacy, you need to test it in non-human primates, period. So this is something that must be done. However, if you want to investigate the function of a gene in a non-human primate, you cannot do this in Europe unless this gene is associated with a devastating, very serious disease that might be life-threatening. So this is the restriction that we have in Europe, and this is also losing the competitiveness, because basically it's providing some competitive advantage to our competitors in Japan, in China, and in US, that they don't have these restrictions. So the message here is that if you can do the experiment in Drosophila or in C. elegans, please do this in these animals, that they are not considered even animals by the law, and do not use pets. If you have to use beagle dogs, that is for developing gene therapies, metabolic disease, you have to use beagle dogs, then it is a mandatory use. Reduction is the second R, is you need to use the minimum number of animals, but you need to keep breeding. Minimum number of animals that allow to reach significant conclusions. If your number of animals doesn't allow to reach significant conclusions, statistically significant conclusions, basically you will be wasting the animals. So this is to avoid repeating experiments. That means you have to prepare your experimental design. You have to take into account the adequate number of animals. Examples of this is this international mouse phenotyping consortium that is creating, similar to what you have in Arabidopsis. It is a database on mouse embryos with mutations in every single gene. And these embryos are kept frozen and are distributed by repositories such as the EMMA. EMMA is the European Mouse Mutant Archive. We are part of EMMA here at the CMB. We are the Spanish node. And here, basically, any researcher that is interested in a given mutation in mice, they don't have to recreate again the same mutation. They basically request this to EMMA, and by doing this, they will be saving animals. How many animals? Well, you see, in order to maintain a mouse colony of a given line live in the shelf, taking into account every 2.5 months, 3 months, you have a generation. Basically, it means that you need to maintain about 50 individuals per year. We have more than 8,000 lines that are frozen in EMMA, which means we're saving 400,000 mice per year, just because we have all these lines frozen. If we did not have these lines frozen, if we had to maintain these lines alive, we will be wasting more than 400,000 mice a year. This is a significant contribution to animal welfare by cryopreservation. We also need to take into account what is the variability between the experimental and your control groups. And if the experimental is very close to the control, you have to increase the number of individuals, logically. And if the experimental, such as in the second case, it's very different from the control, you can use less animals. And there are some equations that you can use to prepare the sample size. Example of reduction. When we started cryopreserving, we were using lots of animals and it took us 1.5 months. Nowadays, with better methods, we only need 2 to 5 males and 2 to 5 females, and we do this in 1 day. So this is an improvement. For instance, CRISPR, you know this in plants as well. In order to have a triple mutant mouse, and you have a double mutant mouse, before we would be creating a third mutation independently, and we will be breeding and we will be making all the crossings, but it will be a nightmare and it will require lots of animals. Now, because of the efficiency of CRISPR, we can already modify the animal that is carrying the 2 mutations, and the animals that are born are already born with the 3 mutations. And this is reducing at least in 60% the number of animals. Refinement, which is the third of the R's, is to use always the most advanced methods. Advanced with respect to animal welfare. So no advanced with respect to the PI. For instance, we're doing embryo transfer very often, and we are anesthetizing the female, we are sterilizing the area, we are opening the skin, we are opening the muscle layer, we are opening the peritoneum, we are exposing the uterus, we're making a hole, we put in the capilla. All this is very mechanic and it's apparently very easy. Easy for you, but it is a serious surgical procedure for the female. You need to stitch all the different layers. You need to provide post-surgical analgesia. And what in alternative, you can do this non-surgical embryo transfer, which is exactly what is being done to women in reproductive clinics. You use an speculum and a flexible capilla to just put it in through the vagina, and basically with a flexible capilla, you deposit the embryo inside the uterus. There is no surgeon, and this can be done in seconds. Of course, you need to learn how to do this, and it's a bit more difficult for you, but it's more pleasant for the female, and it's not creating a surgical. Another example, anesthesia. You can provide anesthesia by doing a intraperitoneal injectable anesthesia. That's very easy. That will provide you the surgical plan for 20 or 30 minutes. But if you are slow or if you need more time, the animal will start awakening and you will be tempted to give more dose of anesthesia, but this will be too much and you might kill the animal. So the best way is to use inhalation anesthesia, so gas, isoflurane, which is what we are given when we go to hospital. Well, the small difference is that injectable anesthesia is very cheap. You only need the syringe and the solution. With inhalation anesthesia, you need to purchase a whole equipment that is provided. But this is something that you have to consider because this is important, because it will be doing a better preservation of the animal welfare. And Clarice, you have a question.
Yes. So I am wondering if there is any entity that goes to the labs and check if everything is going according to plan?
Absolutely, I'm
not?
coming to this. I'm coming to this now.
Okay.
Okay, so basically, absolutely. So basically, you can replace mice with embryos, and you can do the experiments on embryos, and basically you can save animals. So you can use next generation sequences to investigate the founder CRISPR animals and to find out as quick as possible which one is carrying your allele, or you have to find this to follow these ARRIVE guidelines. So this is what Clarice was asking. How do we regulate this? Well, we have this directive, this is 2010-63, that is being transposed to every single state member state. In Spain is the Royal Decree 53 from 2013. And basically what it says, this directive, is that it's an important step towards achieving the final goal of full replacement of procedures on live animals for scientific and educational purpose. But you need to keep reading this, because eventually you have to stop using animals, but at the end of the phrase it says, as soon as it is scientifically possible to do so, which means today is not yet possible to do so. Therefore, we have to be careful when we hear these campaigns that people say there is no need for animals anymore for biomedical research. Well, I wish this would be true. This is not true, and we still need to use animals, but we have to justify this very peculiar. And what these laws they want to maintain is that these laws and the European directive in Spain and any other member states, it is a set of regulations aiming to permit animal experimentation only in registered centers by competent personnel that are adequately trained, educated, and after obtaining the corresponding authorization. So this is to respond Clarice. So in general, what the directive says is that a given researcher needs to validate their proposals first at the level of their institution. Normally it's a local committee that will be evaluating the ethics issues. And when this is being validated, there is this competent authority that will be authorizing. And this competent authority is not only authorizing, but is also auditing. And it is also inspecting every other while this installation and to see whether whatever the permissions were obtained are fulfilled. And this is in general in Europe, and the competent authority can be, such as in France at the national level, there is a unique competent authority, or as in Germany or in Spain, in Germany, Länder, and in Spain, the autonomous governments, the autonomous communities are the ones that have the authority. In Spain, we have a peculiarity that I believe it's not present in any other country, is that we have a third organ, it's the habilitated organ, which is in charge of the project evaluation. So to make things even worse, a researcher in Spain that wants to investigate with animals, first they need to check with the local committee, they need to get the approval from this independent organ that will evaluate, and they need to get the permission from the competent. This normally takes 3 to 4 months, which means and illustrates that it's not very easy to work with animals. And this is what the society requests, and we just have to fulfill. So can we reproduce all experiments using cells in culture? Well, probably not. I'm investigating vision, and I cannot replace the complexity of a human retina in vitro. I mean, there are models that are approaching this, but I still do not see or do not know how to test these artificial retinas to see whether they see an object or whether they don't see an object. So this is why I still need to use animals. Of course, there are improvements, and we have 3D bioprinted organs, and we have organoids, and we have genetically modified animals that have been used to produce a variety of different models for diseases. And this is because we share most of the genes. These are the 23 chromosomes, pairs of chromosomes we have in the human genome, and there are the 20 chromosomes that the mouse genome has. And if you follow the code color, you will see that almost every single piece of the human genome is represented, and sometimes with with very long stretches of genome that is represented in the mouse genome. This is why we have been creating plenty of lines that have been instrumental to understand. And this is why, for instance, I'm using since many years mouse models to investigate rare diseases, in particular albinism, which is the rare disease I'm investigating that is caused by mutations, not in one, but in 22 different genes. Therefore, there are 22 types of albinism. So what about the social perception, which is also an important part of the ethics that is associated with animals? Well, this is always problematic. And as you know, there are companies that are trying to manipulate the public opinion. This is a company selling cosmetics, and they are always collecting signatures to stop using animals from being tested for cosmetics. And would be a nice idea if it would not the case that this is already happening in Europe. So they are cheating. They're not telling the people that in Europe, fortunately, from 2004, 2009, and 2013, we have not one, not two, but 3 European directives prohibiting the use of animals for testing cosmetics. So this is already done. You don't have to sign for it. So there's still some parts of the chemicals, they're not cosmetics, that they are still tested in animals, and this has been now being reduced. Of course, there are people that they are using these very emotional pictures to prevent the use of animals, and these are emotional because they're using baby dogs and they're using this blood and etc. Well, we need to understand what is the philosophy behind these groups, because that's the only way in order to progress into the understanding. And the main question is where do you put the human beings? So do you consider the human beings have a moral status that is different from the rest of animals? This will be my opinion. I think the human beings, they have a higher moral status than the rest of animals, which doesn't mean that I can do whatever I want with the rest of animals. It just means that I have a higher respect for the moral status of human beings. For most of these animalist groups, they include the human beings at the same level of the animals. And so this is the original idea why, if you don't want to do any harm to any other human being, because I consider that the dogs are at the same level, I don't want to do any harm to a dog. So this is to understand what is the principles behind most of these animalist groups that are associated with some ethicists, such as Tom Regan. He was the one writing about animal rights, or Peter Singer, that is he wrote a book on animal liberation that is normally being used. And of course, every other while we get this footage, we get these videos that are recorded in institutions in which they are contravening the norms and they are mistreating the animals. And of course, this is not to be happy about this. I mean, I'm not happy about this, and I only want to say, whenever this is happening, please apply the law and please investigate. And if these institutions are doing things that they shouldn't be doing, they should be penalized, and they should be punished, and they should get a fee, and they should be canceling their own. So, but not because sometimes these institutions are doing these things, the rest of us, we need to be penalized. I'm sorry. Most of the research institutions, they're doing the right thing. We need to face every other while requests for stopping the vivisection. And this is what happened in 2013, when more than 1 million signatures were collected, and they won the right to defend their position before the European Parliament, and they wanted to close down the directive. They wanted to stop the use of animals. And eventually, 2 years later, the Commission responded, and the proposal was rejected because they considered it was still premature to ban on animals, and they wanted to promote these alternative methods. What was the response from the research community? It was dispersed. We are not very well organized as the animalist groups, which are very well organized. There were some institutions or some associations being created, such as by the Basel Declaration, Animal Research Tomorrow, or Understanding Animal Research in UK, or the European Animal Research Association. What we did in Spain, using the Federation of the Spanish Scientific Societies, we created a group that was producing the first white document on the use of animals. We did not explain to the public opinion why we were using animals. We consider that we took it for granted that it would be easier to understand. Nothing is easy, nothing is automatic. You have to explain why you're using animals in research. And then we follow the model that it was launched in UK. They launched the Concordat on Openness on Animal Research. Nowadays is for more than 120 institutions. And what we did the same here in 2016, 2 years later. And we are very happy to say that today we have 163 Spanish institutions that are adhered to this transparency agreement in animal experimentation. Basically, there is 4 points that the institutions that are adhering, they are committing. They're committing to speak with clarity on how, when, and why we use animals, to provide adequate information to whoever is asking, to promote initiatives to generate a better knowledge, and to inform annually. So this is the cloud of logos. So this is, of course, CMD is here and CSIC is here. So I'm proud to show this because it means that we have been able to reach out and to transmit the relevance of going transparent. So basically, we have research centers, societies, universities, private companies, and etc. No? There is a web page. You can see the different institutions and what happens in the rest of the world. Well, today we have 9 countries with similar transparency agreements. First was UK, second Spain, Portugal, Belgium, France, Germany, the Netherlands, then New Zealand, and last was Switzerland. If we take into account all the different institutions, 1 every 3 institutions of the world that is opting for transparency, it's a Spanish one. So this is already showing how we are committing to this. How do we practice transparency? Well, basically we practice by just telling the population how do we use animals and to explain the numbers that the Ministry of Agriculture, Fisheries and Food is providing every year. For instance, we had a very peculiar case last year when all of a sudden in 2021, there was an increase in use of animals. Why was that? It was that because there was a project using feeding larvae from fish, from a given fish species, and that was sort of destroying the statistics because it was more than 500,000 of these larvae, and according to the law, they need to be accounted one by one as if they were non-human primates, and this is nonsense. But this is why we had this increase. When we remove the fishes from the numbers, you see that we are progressively using less animals with the rest of the years. So let me stop 1 second because I want to jump because I want to go to the other topics before I'm using this. Okay. All right. So basically, sorry for this, but I wanted to touch on a different topic. Okay. So basically. So after animals is non-human, non-European countries, and here basically the message is you cannot go to a country that is more favorable for doing the experiment that is prohibited in another country. So what is prohibited in a given European country is prohibited in the European Union. So for instance, you cannot destroy a human embryo for creating embryonic stem cells. In South Africa, you can do this without any problem, but you cannot get money from European Horizon Europe project if you include South Africa as a partner, because basically South Africa will be expected to fulfill the obligations of the ethical standards of Europe. So this is important. So in non-EU countries, you need to be respecting the ethics common standards that we have here. Regarding environment, health, and safety, of course, we have these biological risk agents. I already told you about this BSL 1, 2, and 3, and 4. And just for our understanding, so biosafety level 1 is when you work with E. coli or with yeast, with Saccharomyces cerevisiae, or with mouse cells. BSL 2, biosafety level 2, it's for human cells, for influenza virus, for lentivirus, or for adeno-associated. BSL 3 is this immunodeficiency, human immunodeficiency virus, HIV, or for the SARS-CoV-2, or the SARS-CoV, or the MERS, or the coronavirus that will fall in this category. And the real serious ones for BSL 4 will be the Ebola virus or the smallpox virus. So these are the viruses. Regarding genetically modified organisms, sorry for this being in Spanish still, we have 4 different types depending how serious we might cause disease or we might cause alterations in other animals or in other plants in the environment. And according to this, there will be activities without any risk, or there will be activities with very low risk. There will be activities with moderate risk or with high risk. No? And this is, for instance, what is happening today with the genome-edited organisms. You know that this sentence from the highest court in Europe in July 2018, it was making this correspondence between GMOs that should be treated as of the genome-edited organisms should be treated as GMOs and should be treated as any other transgenic. So, which doesn't make sense, because a transgenic is when you add a transgene, you add an extra piece of DNA. We understand what a transgenic is. We understand what a variety is, in which you might have different polymorphisms associated with a given gene, and we are not calling them transgenic. That's why we should not be calling transgenic when we are moving using CRISPR and we are implementing these polymorphisms. So this is why a genome-edited organism is not a transgenic organism. That was a mistake, but this is what the law has been applied. Further on, because you can get different ways of creating a mutation with radiation, with chemicals, with CRISPR first generation, or with base editors. No matter what you do, you will end up with plants, in this case, that will be indistinguishable. And this is also something that you cannot handle. So, and then we need to change, we need to adapt the law to take into account. And this is the lack of the rational argumentations, because what is happening nowadays is that the decisions being undertaken are not science-based. We consider that these plants that have been mutagenized extraordinarily in the 50s and the 60s with radiation or with chemicals, the ones that we find today in the supermarket, they have plenty of mutations. We have no idea of which mutations they are. But since we have been consuming this since years, they're considered to be safe. When we are crossing, we also bring into the resulting hybrid or resulting variety not only the threats that we want to preserve, but other threats. So we have no idea what other things are traveling. When we're doing gene editing, we know what gene we're modifying. This is the most precise method to this moment, but curiously enough, these are the ones considered to be dangerous. And this is the nonsense situation that has been denounced by many different institutions. And we hope, besides this first report that was produced by the Commission in April 21, we hope that the current directive will be updated, and we will be able to change this current map in which New Zealand and European Union are the only parts of the world in which we are regulating these genome-edited organisms as transgenic as a GMO. So this is how you see the ethics has implication in the biotechnology, in how you conduct your projects and how you would be scaling up your projects from the academia, from your bench to the fields, and how you will be able to set up companies and etc. Artificial intelligence, briefly. This is a new topic. It wasn't there before. And we have to recognize that plenty of projects nowadays, they have artificial intelligence directly or indirectly. There is no law so far. We have recommendations, we have frameworks, we have this high-level expert group on artificial intelligence, which are guidelines that they need to be fulfilled. There was a meeting in Asilomar in 2017 that was recognizing the ethics and values of the artificial intelligence and with respect of human values, personal privacy, the shared benefit, the human control. If we're using a given conversation with someone at the other end of the telephone, we need to know whether this is a human being or whether this is a machine. And we need to be able to cancel the conversation and to cancel the operation of this machine if we don't want to be interacting with this machine. There have been some proposals on some laws and rules and some texts that have been already pre-adopted by the parliament, but as I said, at the moment, we don't have much been already produced. In 2019, there was this ethically aligned design for autonomous and intelligent systems. And these are all guidelines documents that they need to be taken into account, but they are not yet mandatory. So we are still in a process, in a transitional process. Okay, what do I mean when I mean artificial intelligence? Well, this is Peppa. You probably know this is a robot that was built by a Japanese bank that is now being used in museums. It is not very frightening, it's very friendly, and you can interact with him through this tablet. And it speaks very different languages, and it's kind of a very friendly artificial intelligence. Well, this is not so friendly. And this is another robot that was constructed by Boston Dynamics. And this is an example of dual use. So this kind of beast, it can be used for detecting people in an earthquake that has been covered by stones in a given city. So that would be a beneficial use. But it can also be used to bring a bomb to a given place when you are at war. So basically, this is also a dual use. And of course, we can use robots that can be very powerful and can be causing damage to people. And you might think, what has to do this with science? Well, this is the same robot arm in an animal facility that is sort of cleaning cages. And this robot arm is moving very fast. And of course, when this is in operation, no one should be around because otherwise you will get touched and you will be hit by this arm. So there are ethics issues about this association. And of course, to mention the OpenAI, the ChatGPT. So we need to be aware, we're just discovering these natural languages. We're just discovering the potential. And just want to highlight, I don't have anything to do with Microsoft, but I want you to be exposed to this next release of Office package, which is called Copilot. I just want to say like this, you can go and find out videos and you will see what is capable this next version of the Office, which we are all using for preparing our manuscripts. And you will see how artificial intelligence, I will give you just an example. You have 4 or 5 PDFs, papers that have been published on a subject, and your supervisor tells you to prepare a presentation of these 5 papers. So basically, what you do is you start reading the papers, you obtain the images, you produce the presentations. It might take you some time. Now with Copilot, you basically throw the 5 PDFs into PowerPoint, and after a few seconds, the PowerPoint is being created, highlighting the messages, the take-home messages, selecting some pictures, obtaining pictures from other sources if they were required. So basically, it creates a presentation for you. And this is something that we need to be aware. So this is going to be happening whether we like it or not. And this is something that I wanted to at least mention. I have 4 minutes left, and this will be the minutes that I will use for at least mention new ethics challenges. So these are challenges that we didn't know before, but they're just happening now. Chimeras. What do you mean by chimeras? We might be mixing pluripotent embryonic stem cells from rats and from mice, and we are creating a rat mouse or a mouse rat. This is possible. The people that they are considering to mix embryonic stem cells from humans and from pigs, and you will be creating a pig that will have parts of their body that will be human. And the idea is to create pigs that will have organs that will be humanized to be used for transplantation. So, but when do you stop doing this? So what percentage of neurons do you have to take into account before you stop calling this animal a pig and you start calling this animal a human? So this is things that we don't know at the moment, and we need to anticipate it because they will be happening sooner or later. Synthetic embryos. You're probably aware about some results from Jacob Hanna, a Palestinian researcher working in Weizmann in Israel. They were mixing up different embryonic stem cells in mice, and in some certain conditions, they were obtaining embryos, pre-implantation embryos and post-implantation embryos. So there is no longer the unique case that in order to obtain an embryo, you need an egg and a sperm. Now you can get an embryo skipping the fertilization step, and you can go directly and to reconstruct an embryo of a given age. So they went as far as 8.5, 9 days of gestation. I remind you that in mice, it's about 19 days. So it's close to the half of the gestation time. And they have produced an entity that we don't know how to call it. We call it nowadays synthetic embryos. If you do it with mice, now you can do it with humans. And you can create humans whose organs will have the same genetic identity for the person that has been provided these embryonic stem cells. So how do we handle this? You probably saw the movie The Island. So we are getting close to the island. The extinction of species. So George Arches wanted to de-extinguish the woolly mammoth using the similarity with the Asian elephant. Well, for what purpose we are extinguishing these species? He says that he wants not one, but hundreds of thousands of woolly mammoths walking in the tundra in Siberia and compacting the soil and counteracting the ice that is being melted and is being escaping to the atmosphere at the moment. So this is a speculation, but these are happening. Or CRISPR, we know CRISPR can be used for curating disease, for gene therapy, but there are people that they want to use CRISPR for acquiring other capacities, for improvement purposes. And these are an euphemism or something that has a very bad reminiscence and very bad memories. And I'm referring to eugenetics. And this is something we need to take into account. And finally, last but not least, and with this I will finish, the ethics issue with CRISPR in animal biotechnology. Some of them, such as the ones you apply in plants, so to be resistant to plagues, to be compatible with drought, to be more productive. These are things that we can understand. But there is a company in America that they want to inactivate the genes in the cat genome that are coding for the protein that is producing allergy to humans. And this is a protein that is in the saliva of the cats, that the cats is distributing in the body and is touching all the objects that the cat is passing next to. And this is a way that they use to communicate between cats. And basically now we are saying we can use CRISPR to inactivate this gene to produce a cat that will not be allergic to humans. But this is for the benefit of the cat or for the benefit of the humans. So there is clearly an ethic issue that we need to investigate. And so these are the challenges that we need to take into account that were not considered before, that we will have to evaluate, we will have to assess, and we will have to decide how we handle. So I stopped here, and this was kind of an overview on the different ethics aspects that we have in research. And now we can have a half an hour pause. And what I suggest is that we can combine the questions at the end of the second part with the research integrity, which you will see that they are related. Vicente, you agree with this? So
yes.
we do the pause now, 30 minutes. And unless there is any immediate question that you would like to ask, if you have any immediate question, and if you don't, then please don't close the communication, don't close the connection, simply switch off the cameras and switch off the microphones. And at 12 at noon, we will be reconvening and we will be resuming. All right?
Okay. Thank you, Luis. So see you in 30 minutes.
See you in 30 minutes, everyone.
Igual que estas. Ana, ¿cómo estás?
Dame un beso. ¿Qué
¿Cómo
tal?
estás? ¿De tu afección?
Jo,
¿Mejor o qué?
yo creo que mejor. Yo creo que las medicinas
¿Crees?
han hecho su efecto.
Siempre.
Lo que pasa que...
Sí, mira.
Sí.
Justo a la misma altura que estamos. A la de...
Ana,
O sea, que de vez en cuando me da el ataque y no paro.
Ana, Ana,
¿Tienes caramelos? ¿Tienes caramelos?
No, de hecho te iba a coger, digo,
Pues
porque
coge todos los que
me coges una de... una-
Ana, Ana Carmen.
O sea, acaso me da la tos.
varios.
No, hombre, ahí tiene que haber que todo el mundo.
¿Eh?
Ana.
La gente no quiere eso.
Pues yo por la garganta.
Voy a llamar por
Me
teléfono.
un chisme así en jarabe, ¿eh? Y me lo he echado en un bote pequeño.
¿Tú quieres llevarte, pregunta ahora, la taza ahora o quieres llevártela? Yo no te lo digo, pues. ¿O la quieres ahora?
Si luego me voy a ir de... O no, me la cojo mañana. Así no cargo con ella.
Ella como tú dices. La idea es porque, claro, la tienes que llevar en la bolsa todo el día. Ahí como tú quieres.
Como mañana, pues te digo, porque mañana...
Y te quedas a todo, ¿no?
Sí.
Mañana comes también.
He cogido 2 días de vacaciones, aunque estoy trabajando. Mira, me está llamando Vicente.
Porque no lo solucionan.
Okay, we will start in 1 minute. So 11:59 in my clock. In 1 minute, I'll start resuming the class.
Okay. See.
All right.
Okay. Maybe one or two are missing.
Oh, we can start and then people can sort of join in.
Okay.
So let's get started with the second class. Okay, this will be on research integrity. And this is something that I particularly care because I assume that not many of you have received formation or education on ethics in research as we did in the first class, but I would probably assume even fewer of you have received formation or education on research integrity. And these are fundamental aspects that are transversal, that are horizontal to all the different disciplines, and they need to be learned. So it is not that some people, they say, well, this is common sense. Well, you have to behave adequately. Well, besides behaving adequately, you need to follow some rules. And these are the rules that we will be discussing in our profession, that are these codes of conduct that regulate what we're doing. Maybe to begin with, maybe you can tell me, maybe you can activate the camera and tell me who has received education on research integrity, if any. At least I see two people, Shannon and Clarice. They say that they have received education on research integrity. Shannon says yes, and Clarice says, I don't know.
I think I didn't.
Okay, so but Shannon, you received something, you said, right?
I did bioethics as an undergrad course.
But that includes research integrity as well?
A little bit, yeah. Okay. But it was never a full module ever.
Okay, fantastic. But at least we have someone, so I'm glad. I'm glad that at least, but for the rest, I understand that you have not received it, and I understand this is why I acknowledge and I appreciate Vicente's efforts to have you here for this second class. Well, research integrity, it's about many things. It's about honesty, it's about intellectual property, it's about being a good professional, it's about authorship, it's about codes of conduct, it's about ethical behavior. But basically, just to put this thing very clear, this is the elephant in the room. So this is the elephant in the room that nobody wants to see. But the elephant is there. And the elephant, if you don't pay attention to this, will continue breaking the tables and breaking the chairs. And sooner or later, you will have to take into account that you have a problem. And this is the problem that needs to be anticipated and needs to be well handled through the different codes of conduct. So research integrity is an essential part of what we're doing. So basically, it's this endless battle between the angel and the devil. The angel will say, don't cheat, don't lie, don't fabricate, follow the rules. And the devil will say, cheat, copy, fabricate, don't follow the rules. Okay, so this is something that we need to take into account. So often science is presented as a creative profession in which basically, apparently, we have no rules because we can do research on whatever we want. Well, this is not true. This is the first thing you need to learn. In science, we have a framework that delimits what we can do in science. And outside this framework is things that we cannot and we should not be doing. So this is this deontological codes that regulates the profession, each profession, whether you are a taxi driver, a flight pilot, or whether you are selling oranges in a market, or whether you are a politician, or whether you are a judge in the court. There is always a framework that delimits what you can do in your profession and what you should not be doing. Therefore, it is not true that in science we can do everything we want. So research integrity refers to honesty, conveying information truthfully and honoring commitments, accuracy, reporting the findings precisely. And precisely means not skipping results and not exaggerating the results. Efficiency, using the resources which are always limited wisely and avoiding wasting them. And objectivity, which let the facts speak for themselves and avoid improper bias. You're doing experiments to confirm hypotheses. You're not doing the experiments to confirm your idea that is pre-existing, that you want to demonstrate. No, you are not demonstrating an idea. You are demonstrating hypothesis. And if these facts tells you that your idea, your hypothesis was not correct, you need to change the hypothesis. You don't have to change the facts. You don't have to change the results to force them fit into your pre-existing hypothesis. Research integrity is intrinsic to all our research activity and excellence because it's really at the core, because it's the record that we are producing, and that's the basis on the society trust in research evidence and expertise. Basically, what you're producing is not only for you, for your PhD, for your career, but it is also for anyone following you that will be reading your papers and will be basing their new hypotheses on the facts that you were reporting at the time. And if you were committing a breach in this research integrity during your papers, you will be creating noise. You will be adding noise to the registry, and whoever would be try to follow and try to reproduce and try to repeat these experiments will not be able to reproduce these experiments. And this is definitely a main problem that we have to solve. Research integrity safeguards the foundations of science and scholarship and maintains public confidence, underpins the continued public investment and protects the reputation and careers of researchers, prevents adverse impact on patients and the public, promotes the economic advance, and prevents the always avoidable waste of resources. So there is a lot to be charged on the shoulders of the research integrity. So this is why it requires a module. This is why we are devoting 2 hours to this topic this morning. So research integrity might be dissected in 2 different aspects that I will be presenting today. First is about credibility, trustability. And this trustability relies on the quality of data that we are reporting and regarding the reproducibility of the results that we are reporting. If we're finding something unexpected, we publish, but no one else is able to reproduce, people might think that we might have another alternative explanation for these results, not necessarily being scientifically based. We might have forced this data. We might have forced the experiment or pushed the experiment so that the results that we obtain are the ones that fit with a idea we had in mind before. And basically we're doing these experiments to confirm the idea, whereas the experiments are always to demonstrate the hypothesis that should be always first. First the hypothesis, second demonstration experimentally of the hypothesis. And if the hypothesis cannot be demonstrated, we change the hypothesis and we start again. It is called the scientific method. And this is it should be engraved in the stones of the science And this is the basis of the research integrity. And the second aspect is that you need to learn how to behave appropriately. And as I said, this is beyond applying common sense. This is the so-called the codes of good scientific practices that every institute should have. First of all, what I would say, you should investigate in the institution that you work in, whether they have or not a code of good scientific practices. Normally, all institutions, they should have. And if not, there are European codes that apply to all of us. And particularly, since you are receiving European money because of these Marie Slowodska-Curie actions, you are obliged to fulfill and to comply with the European Code of good scientific practices that I will be referring next. So even if you think you don't have a code, you do, because you are receiving European money for your research. The Singapore Statement on Research Integrity also refers to similar aspects of honesty, accountability, courtesy and fairness, and good stewardship. So there are different aspects that basically are referring to the same issues. Where to find introductory information on research integrity? Well, there's a number of reports that are available in the internet that I strongly recommend that at least you browse. Ideally, you should read them. This is from the National Academy of Science in the States. That's the 3rd edition of a very good guide for responsible conduct in research. This is called On Being a Scientist. And this I strongly recommend. Also, we have the Office for Research Integrity in the NIH in the US. They have the Introduction to the Responsible Conduct of Research. That's a rather old guide document, but still most of the contents will apply today. So 20 years later, this book, it's still good to be referred. In 2010, in Europe, we had the ESF, the European Science Foundation, was producing this document after one meeting day it was being held on that year. And this is about fostering research integrity in Europe. And what I was referring before, this is the code of conduct that applies to all of you, because you are grantees of this MSCA action. And the MSCA action are bound to the European Code of Conduct for Research Integrity. The last edition is 2017. You can get them into the ALEA. ALEA is the All European Academy's website, alea.org, and you can get this European code, and you can get them in all the different European languages. So there is no excuse. So basically, if you have to choose one document to read, I would say you should read this, because that's the one that is governing your attitude, that is governing your behavior, no matter where you are in Europe, no matter in which institution you are. This is, for instance, the Spanish version of this code, but you have the French, the English, of course, the German, the Czech, the Italian, the Danish. I mean, you have all the different languages. The European code relies on honesty in communication, reliability in performance research, objectivity, impartiality, openness, duty of care, fairness, responsibility. You see that these nouns and these adjectives and these aspects, these values are being repeated all the time. In UK, you have an universal ethical code for scientists with the 3 R's, in this case, rigor, respect, and responsibility. And this is what we have in our institution. In the Spanish National Research Council, that we all belong, Vicente and myself, and this is the center from where I'm delivering this talk. The National Center for Biotechnology is one of the 150 centers that belong to this, the largest public research institution in Spain. We had a previous code that was produced in 2010 and published in 2011. It was already published in Spanish and English. And 10 years later, and I'm proud because that was occurring when I was chairing this committee, this ethics committee of the CSIC, we released the second edition, which is the 2021 edition. And this is the CSIC Code of Good Scientific Practices, taking into account, for instance, items that were not covered before. For instance, the relationship between the researchers and the mass media and the communication media and the social networks. And this is important because during the pandemic, many scientists were approached for answering questions. And one has to remember one important thing. Both physicians and researchers are the two highest categories that deserve the highest trust in the society nowadays, which means that when a researcher, when a man or a woman researcher is being asked on a given topic, whatever he or she would say will be considered as the truth. So you have a responsibility when you're speaking in front of a radio or a television program, or when you are responding to a journalist, because you need to be carefully selecting what you're going to be saying. If you're an expert in that topic, you might as well deliver your opinion. But if you are not an expert and the level of knowledge you have about that topic is just the same as any other citizen that would be informed through newspapers, you should not be commenting. You should let these comments to other people that are more expert and are more familiar with this topic. And this is, for instance, one aspect that wasn't in the 2011, and it's present in the 2021 edition. You can get a copy of this edition. Now, if you take your telephone and you scan this QR code, you will go directly to the page from which you can download this code. And I would just sort of recommend you to browse it, because you will see that they are very similar. All the codes of good scientific practices that are produced in all the different institutions are similar, but it's good to have a look and to see how are we handling these issues here at the CSIC. We even produce an infographic in which we were sort of summarizing the most important aspects. At the CIC, we also promote in 2015 a national declaration on research integrity. And we did this because we didn't have, at the time, we didn't have any body that would take care of research integrity at the national level. Now we do. Now we do, because the law, we have a new law of science from last September 2022 that includes the Spanish Committee of Ethics in Research. And this Spanish Committee of Ethics in Research, among the aims and among the duties that has associated are to be acting as a national office for research integrity. So we didn't have that before, but we have it now. This is important because this is how is Europe these days, and this is thanks to this map that is being produced by NRIO. NRIO is the European Network of Research Integrity Offices, and the code color means whether such an office exists or not. For instance, we were blue because, like in France or Poland or Ireland, we did not have this framework. We did not have this research integrity framework. Now we should be green. We should be like Belgium, Germany, Austria, or Sweden, or Finland, or UK, in which they have these national integrity, national research integrity offices. For instance, there are even countries which they have no guidelines and they haven't identified this issue as a relevant issue. For instance, Italy, Portugal, and we have other countries, and Iceland, and etc. Other countries in which this is yet to be defined. And at the level of the European Commission, you know that the body that is assessing the European Commission on these ethical aspects is the EGE. The EGE is the European Groups of Ethics. And this is now being chaired by Barbara Preinzak. And this is independent, it is plural, it's multidisciplinary, and it is assessing the European Commission on ethical aspects. I'm telling you this because the image, that the mirror that we should be looking at, so the aim that we should be aiming is to be close to the US Office of Research Integrity. This office was established in 1989, so quite a while ago. I just told you that in Spain, we established this last September, and actually just a couple of weeks, this committee was being appointed and the members was being communicated. What's the role of this Research Integrity Office? Well, this is important in the US because every year they publish the list of people that they have contravened, that they have committed any fraud or any plagiarism or any cheating on their research. And this is important because if a person is being investigated and accused and found guilty of research, of contravening research integrity, this person, the name of this person and the results of the investigations will be published. And if this person wants to be hired by another institution, this new institution has the right to double check first at the ORI and to see whether the person they want to hire, it's being clear or it's been associated with some charges that had happened to this person before. For instance, we don't have this in Spain. And if in the south of Spain, someone is crossing the law and creating some problems regarding research integrity and it's being fired from this university, the person can go a couple of weeks later to the opposite corner of the country. And if this new institution doesn't specifically ask this original CV or what happened to this person in the previous institutions, they have no way to know that this person has been investigated and has been found guilty. And this is what is happening in the States. In the States, the Offices for Research Integrity is an executive one, which means this office can apply fines and can send people to jail. Whereas here in Europe, in most of the European countries, if not to say all these offices for research integrity are assessing. They are not executive. They are assessing, they are recommending actions to the dean, to the president of the institution, and the president of the institution is the one that has the capacity to take any decision associated with the investigation that is being done. For instance, this is Charlestown in 2020. You click on Charlestown and it says that has found research misconduct, intentionally, knowingly, and recklessly falsifying or fabricating data, including in the following 6 grand applications and including in so many papers and in figures and et cetera, et cetera. So you have a clear information what this person has been crossing. Okay, why the science is going wrong? Why do we have a problem on research integrity? This was an exercise that was published by The Economist in 2013, and that was followed by a test on reproducibility that was originally organized from Nature in 2016. Almost 1,600 researchers were asked whether they thought that they were facing a reproducibility crisis or not. And if you pay attention to the red, it basically says that 90% of researchers thought there was a significant or it was a slight crisis of reproducibility. Only 3% said there was no crisis and 7% said they didn't know. So clearly we have a problem. What is the type of problem we are facing? And does it depend on the different topics, on the different areas? Well, reality is that the problem is transversal to all different areas: chemistry, physics, environment, biology, medicine, others, and etc. And when you try to categorize what is the source of this lack of reproducibility, the first one is always, if you see the graphic on the right hand at the bottom, it says selective reporting. This is cherry picking. I'm only reporting the things that go along with my idea. I'm only selecting the data from my experiments that favor my hypothesis. I'm discarding those that are contravening, that are going against my hypothesis. This is the main problem that we are facing in science. Of course, then we have pressure to publish, low statistical power, not enough replicas, methods that are not perfectly described, and we have, of course, fraud, and we have insufficient peer review. These are things that are added, but the number one, close to 70% of cases, is selective reporting. Is this, I'm not interested in this data, because this data is making that the difference that I'm looking forward to find between this control and this experimental group, if I include this data, I will not find any statistical significance. But if I discard this point, all of a sudden, this becomes statistically significant. And this, believe it, this is really one of the most important problems. For instance, if I tell you, what is the difference between these two comparisons between the blue and the green groups? So on the left, there is no statistical significance. The P is 0.15, that is considered not statistically significant, and so you are not very happy because the experiment did not fulfill your expectations. On the right, you have a P which is 0.015, which is, of course, statistically significant. You can add a star, and that goes directly to figure 1 of your paper. So what is the difference between these two series of data? Well, the only difference is that on the left, you have taken into account all the points of your experiment. And on the right, for example, you have forgotten or you have discarded just 1 point. And just because you discard this outlier, what you consider an outlier, all of a sudden you realize that this becomes statistically significant. You just don't know how frequent this is. And this is our beast. This is our main problem in research. You should please avoid doing this. And there are some labs in which students are taught this way, in which students are taught to discard outliers because outliers are not informative, because you even convince yourself, because you say, I'm discarding this point because that was when my mom was calling me. I was picking the telephone, and instead of staying 15 seconds, I stayed 30 seconds with this reaction. And of course, this was wrong. So you convince, you try to find excuses to please yourself and to say, this is the point I have to discard. But you see, this is the main problem, because at the end of the day, if you publish the right figure, you will be telling whoever is reading the paper that there is a significant difference between your control and your experimental group, which might be the results of giving a medicament or giving a drug to animals, or to make it worse, to giving a drug to some patients. And you are transmitting the idea that this drug is beneficial, that this drug is functional, whereas the reality is that you have different sources of variability that you are not taking into account. And if you want to establish a statistical significance, maybe you have to increase the number of points in your experiment, or you just have to assume that there are no statistical differences and that your experiment is, you have to change the experiment because the hypothesis has not been demonstrated. So I'm spending some time in this because this is the main problem. Whenever I'm teaching about this, I always see people moving in their seats in the room, because the people starting to talk to each other, because some of them, they say, well, I saw this all the time in my lab and nobody told me that this was not something that I should not be doing. Well, now you know that this is the main problem you have. What are the other problems that we're having? Well, there's many journals that are offering you to publish almost everything you want. These are the so-called predatory journals and predatory publishers. And there is always, we can even spend some time discussion on this. And basically, even if you publish rubbish, this rubbish, you can publish, and we will get a record, and this will be cited, and people will be believing, and these are often without the adequate reviews and etc. So this is adding also noise to the registry. Why research misconduct, vulneration, this contravening research integrity is wrong? This is what we discussed in an EMBO course in which I was also teaching on this, because it adds wrong or useless results to the scientific record. This is the main problem. We are telling people that will be reading these results that these are true results, and they are not true. They're wrong, they're useless. We're wasting resources, we are reducing public trust, we are endangering public funding for the future if we are found to be guilty of this, and we might be endangering people's lives if we are dealing with a drug or a therapy, and we could impact negatively on the policy and the legislation. So all is negative, and we should not be doing this. An example, this is a crystal clear example, why research misconduct is wrong. This is the top 4 papers that were highly cited and were retracted. And there are 4 papers in 4 very respectable journals: New England Journal of Medicine, Science, Lancet, and Plant Journal. And I'm listing the year in which they were published, the year in which they were retracted, the reference they had before retraction, and just to make it worse, the reference they accumulate after retraction. So basically, pay attention to this Science paper from 2005. They apparently had only 200 and so references citation before being retracted, but after being retracted, because the paper doesn't disappear, it's only labeled as retracted, and because people is not reading the papers, but is citing these papers according to the title or according to previous citation exercises, there's more than 1,000 papers in which this paper has been cited again, amplifying the negativeness, amplifying this bad reputation, amplifying this bad noise, this this problem in the literature. Another way of doing research misconduct. This is, for instance, during the pandemic. During the pandemic, you probably know that there was this paper that was published in March 2020 by some French immunologists from Marseille. And just to give you another bit of information, the editor of this journal was the main author of this paper, just for you to inform. They were claiming that hydroxychloroquine would be useful to counteract the effects of the coronavirus. And they were using this graphic. This graphic, it's a disaster of graphic, because to begin with, the numbers that I've added to the right, they were not part of the graphic. The graphic is only representing the means of these individuals that were tested, and that represents the percentage of patients that were PCR positive after the treatment, either with the hydroxychloroquine, that is in blue, hydroxychloroquine with an antibiotic in green, or the control group not receiving hydroxychloroquine in black. So the reality, if you read the paper, they say they have 16 control samples. In reality, from 12 of them, they don't have PCR data. They only have 4 controls. For the hydroxychloroquine tested, they only have 14, and for the ones with antibiotic, they only have 6. They don't tell us the error bars. We don't know the variability. But believe it or not, this graphic was the graphic that was used by Donald Trump and by Bolsonaro in US and Brazil just to recommend that hydroxychloroquine to be used. And all of a sudden, there was a shortage of this medicament that is being used by some people with autoimmune disease, such as lupus, that they need to use this medicament, and they couldn't find this medicament because this was exaggerated. And this is the exaggeration was causing. And this was even resulting in two very big papers in Lancet and New England Journal of Medicine. I'm not talking about very small journals. These are the top medical journals that they had to retract coronavirus papers because they were doing fraud on this hydroxychloroquine. This is the retraction of the New England Journal of Medicine, and this is the retraction of Lancet. So you see that even with something as relevant and as serious as a potential treatment for COVID-19 that wasn't a treatment, it was not a treatment. Hydroxychloroquine was never found to be useful for treating COVID-19. So many papers and so many money. Even in our country, when there were some millions of euros devoted to research, the first series of projects were also to fund research on the use of hydroxychloroquine. These were money that was being wasted because there was no scientific evidences on this. And everything was based on a single figure, on a single article that was claiming to have found a cure for COVID-19. And as this happened any other time during the pandemic, well, this is from last month. Perhaps we have a bit more. So today, we have more than 350,000 papers that have been published on COVID-19. So this is a tremendous amount. So there are topics that have been living with us for many years that they don't have as many papers. Okay, how many of them have been retracted? And this is being recorded in Retraction Watch. Well, more than 315. Well, depending how you want to see this is about 1%. This is 1 per 1,000 of the retractions. Is this a big or is this a small number? Well, I think it is a big number because we're talking about more than 300 studies that were not right. More than 300 studies that those people that would be believing, that will be basing their future research on the results published in these papers, they will realize that they will go nowhere because they will be unable to reproduce the results reported by these papers. So you see, even during the pandemic, this misconduct has been impacted in science. Another example on how the research misconduct has been impacted in science. We all know, and I guess you all know as well, because this is what we learned during the pandemic, if you didn't know before, the development of a vaccine or of a drug. We have a preclinical phase. There's always an idea that is being explored in the laboratory, in vitro. Eventually, you go to a preclinical test with animal models. This is the in vivo phase. And if everything goes well, you can start the clinical phase. So the clinical trials, that is phase one. Phase one, you evaluate first safety. Remember the principles of bioethics. First is non-maleficence. First is safety. Phase two, you continue with safety and you start evaluating efficacy, maybe with a reduced number of volunteers. And phase three, you have already decided what would be the administration route and what would be the dose. And then you recruit more patients, more volunteers. And eventually, if everything goes okay, you enter the phase four. Phase four is the pharmacovigilance. So basically, the drug has been approved, but you continue exploring whatever it might be coming from the people receiving this drug. Okay, this is something that we all need to respect. And this is the way, it might take whatever it takes, but this is the way to be sure that we will be producing drugs and therapies that are respectful with the human dignity, that are safe, and that are efficient. Of course, then we have these other situations, and they might be funny. I don't think they're funny at all. I think they are very serious. Here we have one researcher that I admire, I respect them fully. On the right is George Church. And George Church was claiming that in his garage, he could do some incubator and he could produce the specular protein from the coronavirus, the S protein, and he will be able to lyophilize this protein, and he can administer himself this as a natural vaccine produced in the backyard of his home. Well, this is sending a very wrong message. This is sending a message that basically you can do whatever you want. You can skip all the preclinical phase, you can skip clinical studies, and you can basically produce yourself this vaccine and administer yourself this vaccine. This, I thought it was meant to be as an alternative for producing this, but of course, this is really detrimental for all of us that we are following the rules and respecting the process. There was a complication on this that was even explored, and eventually this happened in UK, in which in order to speed up the process of vaccines, they said, what about if we give vaccines to people and then we, in purpose, we infect coronavirus into these people and see how many of them develop the disease? Well, this is, you might think this is a good idea, but you're dealing with a virus that is causing about 1% of deaths in infected people. So this is not a free virus. It's a virus that is causing a respectable amount of deaths. So this is very different as when you do a clinical trial in which, to begin with, you're using volunteers and you are comparing the vaccine with a placebo. You are comparing a vaccine with something that is not providing the vaccine. Basically, the volunteers, they don't know whether they will be receiving the vaccine or some PBS, and they will be released to the society, and during their lives, they might be exposed to coronavirus. And if they did not receive the vaccine, they might be infected and they might die. And those that would be receiving the vaccine, they might be protected and they will not die. So even though the situations might be similar, they are totally different. And this is a reminiscence of something that you need to know, which is the tramway ethics dilemma. I don't know whether some of you have already come across to this, but think about this. There is a tram going without control, and you are in operation, and you can diverge the tram, because if you don't do this, the tram will kill 5 people that are bound, tightly bound to the tracks. But if you operate the tracks, you only will be killing 1. If you ask yourself what you would be doing, I guess almost 100% of you will decide to operate the tracks, and you will say that killing 1 is better than killing 5. Therefore, at least you will find yourself more relief, because this will be something that you think it's good. This is similar to when you are doing the test on volunteers. Some of the volunteers will not be receiving the vaccine, will be receiving the placebo, and because of that, they will be dying because they will be infected. So this is the minimum number of people. This is the 1 person attached to the tracks that will be dying for the benefit of many, for the benefit of the community. Now we see the second part of the tramway ethics dilemma, which is you are in a bridge, you have the same tramway out of control, will be killing 5 people, but you are sitting next to a obese person, a fat person, and you can push this person into the tracks, and by doing this, you will be killing this person, but this person will be blocking the tramway and you will be saving these 5 people. In principle, on paper, it is essentially the same, because you say you are killing 1 person and you're saving 5. But here there is an action. Here you are doing something proactively, and many of you probably will not be pushing this person. And many of you would be assuming that unfortunately these 5 poor people will be dead. So this is similar to what happens when in UK, they want to, in purpose, deliver coronavirus to people. because by doing this, they know that they will be killing some of these people. So you see, this is how you have to face these ethics dilemmas that are happening, for instance, during the pandemic. So what is research misconduct after all? I've already given you a couple of examples, but let's try to define research misconduct. These are behaviors that significantly compromise the proper professional conduct of research, or, and this is important, the accuracy of the research record, either because you are misbehaving or because what you have been reporting is wrong. So basically, this is about falsification, about fabrication, and about plagiarism, not only of others, work from others, but also self-plagiarism of yourself, fabricating data or falsifying data. So this is why the message that is conveyed when you're talking about research integrity is do not lie, do not cheat, do not steal. So people might say, well, this is normally very clear, and this is black and white. So the world is divided between those that are born in Andorra and those that are not born in Andorra. You are born in Andorra, you are an Andorran. You are not born in Andorra, you are not an Andorran. So this is very clear. There is not much discussion. Unfortunately, in ethics, nothing is black and white. In ethics, you have to pay attention and you have to define and you have to discuss and you have to interview the people that have been accused of breaching this research integrity or research misconduct, because not all research misconducts can be explained the same way, and not all research misconducts have to be penalized. For instance, I think we all agree that manipulating an image might be wrong or will be wrong. But I'm presenting you these two pictures. Have a look in these two pictures. Do you see any difference between these two pictures? Well, I hope you see that there is some feces here. So because the mice are nervous, one of them was cheating, and then he have a piece of cheat here, which is not nice. And you have used the Adobe Photoshop, which to remove these feces. Well, you have manipulated the image, but have you changed the information that is conveyed in the image? I don't think so. So the same 6 mice that you were showing before are still being shown. So you probably can solve this by saying these images has been edited with Photoshop by removing items in the image that were not informative for the purpose of the research being demonstrated. But this is, if you are strict, if you are black and white, you might say this is an image manipulation, this is research misconduct. But if you pay attention to the reality, you will say, well, this is something that can be handled very easily. Something else is this. You realize that the picture contains a mouse that shouldn't be there, but you don't have the mice anymore. You killed them all already. And now you apply Photoshop and more or less you try to remove this mouse. And now you are presenting a picture with 5 instead of 6 mice. This is manipulating the image, and this is altering the record, because now you're showing something that wasn't there. You're showing only 5 animals, and you are removing an animal that probably did not fit in your hypothesis. This is a clear research misconduct, whereas the first one was not that clear. Look at these pictures. Do you think these are different images? These are cells in culture being stained with the cytoskeleton and the nuclei. Well, they apparently are different, but in reality they are not, because if you flip them vertically and then rotate 90 to the right, you will realize that it's the same picture that has been changed in the orientation in order to transmit the idea that it was a different image. Well, clearly, even though people might say, well, that was a mistake, clearly this is something that has to be at least considered as a research misconduct. Another example, look at these northerns. I mean, some of you might have heard about Northern. Everyone now is doing RNA-seq. But for people from the age of Vicente and myself, we used to do Northerns. We used to obtain RNA from tissues, and we used to transfer this RNA to nitrocellulose membranes, and then on nylon membranes, and we would use a probe to hybridize. So let's say you're investigating the expression of gene X, and this happens to be expressed in prostate because that's where you have the highest expression, because you have a control RNA. Now you have to repeat the experiment, but you are lazy, and you are repeating the experiment, but you don't want to repeat the control RNA. And then you are reusing this control RNA from a previous experiment, and in order to make things look a bit more different, you flip the orientation of the control RNA. You flip it, and all of a sudden, it is just the opposite, and you claim that still gene X is expressed in prostate. Of course, you have manipulated the image. Definitely, this is a clear manipulation because you are using as a control RNA something that was not the proper control RNA. Further, this has been changed, but you are not altering the scientific record because the conclusion is the same. Gene X is still expressed in prostate. Let's say another situation in which you are lazy again and you decide to flip the second image. And if you flip the second image, all of a sudden, the highest image, the highest impression, the highest signal goes into small intestine. Therefore, now you are altering, you are manipulating the image and you are altering the scientific record because gene X is no longer expressed in prostate. Because you see there is almost no signal. It is now expressed in a small intestine. So you see that there are different levels of manipulating images, which they need to be associated with different levels of punishment. So there are people that they say, well, this is black and white. You are manipulating an image. You have to retract everything. You have to change everything. Well, if you have not altered the scientific record, you might have a chance to explain why this might have happened. You have to apologize. You can provide the better picture or a newer picture. But if you have changed the scientific record, then there is no escape. There is no use. So this is why we are dealing with not with black and white, but with gray. Another example is when you compare these two series. For instance, if you compare the left panel and the right panel, probably if I ask you which one of the two panels you prefer, you will see that the right panel is the one preferred because it has less variability. Well, it doesn't have the less variability. It has the same variability, but you're expressing them differently. On the right is a standard deviation. On the left is a standard deviation. On the right is the standard error of the mean, SEM. And of course, when you are using the standard error of the mean, you know that it's a standard deviation divided by the square root of the number of samples. Therefore, the standard error of the mean will be always lower figure. It will be always a reduced number. Therefore, it will give this impression that you are having a less variability. If you are reporting the SEM, you need to report the N, because if you are reporting the SEM and you are letting the reader know or letting the reader assume that you will be using the standard deviation, you will be misguiding the reader, because the reader might think that this experiment has less variation, less variability than it really has. So there is no problem with you want to use the standard deviation or the SEM. But if you use the SEM, you need to indicate, and in particular, you need to indicate the N, because then you can obtain the standard, proper standard deviation in this experiment. So therefore, this is all to say that depending on how is the level of the alteration, you might put the red line there or there or there. So this is why you need ethical committees. You need committees in which these things are being explored calmly and with due respect to everyone, interviewing everyone that is involved in the case, because otherwise it will be too simple. It will be you have manipulated an image. There is nothing you can do. Basically, we retract the paper. And in some cases, I'm not saying in all cases, but in some cases, you can provide a corrected image, and this will be sort of enough with the corresponding apologies. And so this will be enough to overcome this case. Plagiarism. Well, these are two papers that are from different authors, from different institutions. They are published in different journals. But with the exception of what is in the circles, in the red circles, the rest is exactly the same words. So this is 100% plagiarism. And these are people that are changing one word in the title, and basically the rest of the article is the same. They're using a predatory journal. And of course, this is adding a new line in their CV. But this is totally fake, because they are sort of plagiarizing, they are copying exactly someone else's work. Well, this is also happening to us, and we need to admit this. We are running short in time. We need to send an abstract to a meeting. We prepare an abstract. 3 months later, we need to send another abstract to another meeting, but then we also running short in time. And because we're running short in time, we just send the same abstract to the second meeting. And let's face it, we all have done this. But this is not good, because we are letting the participants in the second meeting to believe that what we are reporting is is our original work, whereas it was already presented in the previous meeting. So we need to adapt, we need to change, we need to do whatever it takes to produce a new text every time for the abstracts. And in principle, we have to avoid text recycling. It is permissive. This is the level in which you start getting into the plagiarism. Around 25–30% of the words and phrases you can still use, because there's only a certain number of ways you can describe, let's say, a given concept in science. And once you have described this concept, so there's no other way that you can describe. So this is permissible. But with this plagiarism, what you can use to detect and what the journals are using to detect are these different applications. This is Plague, the Whipper, the CopyLeaks, and particularly Turnitin, Paperwriter, Plagium. These are, some of them are free, some of them you have to purchase. But I'm normally saying that you don't have to worry much of this, because you can use Google. Google, some of you know already, if you put a phrase that can be as long as you want between brackets, Google will try to find exactly the same phrase somewhere else. And this is probably one of the most easiest and more efficient ways of finding plagiarism when you are selecting a given paper. For instance, this is one paper on my lab, and I want to know whether this phrase that I'm most proud of, for more than 20 years, these systems were of interest only to specialists. This is a genuine phrase. This is a phrase I have produced, or this is a phrase I have stolen from any other paper. So I apply this to Google, and all the links that they will be proposed, they will be bringing me to my only paper. So I'm sure that this was something that I produced myself. With plagiarism, what happens is something very peculiar and very funny. When we are asking people whether they see plagiarism in other colleagues, they see 30% of their colleagues, they recognize they have been applying plagiarism, they have been copying. When the question is applied to yourself, self-admitted is only 1.7%. So let's say you are seeing plagiarism in 30% of the colleagues, but you are only admitting a 2% of plagiarism, which of course is something that should make you think that something is wrong here. Okay, where, sorry for the Spanish, I mean, some of them I left already in Spanish, where this research misconduct has still appearing, well, during the planification, the execution, the interpretation, the publication, and of course during the revision of this, during the revisions of this work. So especially in the revision, because you are friend of the researcher, and then you will be promoting the publication of the results, or you are a competitor, and you will try to find problems, even if there are no problems, just to prevent or just to avoid the publication. So this is also research misconduct. What are the consequences for individuals? Well, this is the end of career, the suspension of job for many. There is decreased possibilities for subsequent funding. It is a discredit. This is the only thing we have is the credit, the trust of our colleagues. We are losing prestige. We might be entering into a depression. And in the US, not in Europe, because remember in Europe, this is not executive, this is only as an assessment. In US, they can have to face fines or prison. This is, for instance, two examples, a European example and a US example. Olivier Boinet from your field, and he was found to be committing research misconduct, a number of papers retracted or corrected. He had to return funds, he had to return awards. He was accused and he was investigated by different institutions. Eventually he was released and he was basically left to go back to research, and he's now back in research and publishing. And this is something that is happening here in Europe. Whereas in the States, this other person, Don Pughan, claimed he had found a vaccine for HIV, and he was cheating. He was fired. He had to return the money he was receiving plus a high penalty, so $7 million US. And he was sentenced to 5 years in prison. So you see, research misconduct in both cases. In Europe, we are more permissive, whereas in the US, it's a serious stuff. Here, what happens in many countries is that all projects are investigated, and this is a tremendous bureaucratic effort. Here, whenever you are receiving some money, then sooner or later you will be audited and you will be investigated. And even if they found something wrong when you purchase a taxi from an airport to the center, maybe 20 euros taxi, and you forgot this invoice or something, then they will make a big thing, even though this is just something, a stupid parameter, a stupid thing. In the US, they don't do systematic inspections. They do random inspections. But everybody knows that if you are found to be guilty of research misconduct, you will have to face serious consequences. And it is enough to know this so that most people will follow the rules and will not be tempted to commit this research misconduct. So these are recent papers of Boinet, so he's back in science. Examples in the literature about research misconduct or claims for research misconduct. You probably remember, or you have read or studied, that in February '97 is when we knew about Dolly the sheep, the first cloned animal. And the front page of Nature was this picture in which that was generating a lot of controversy, because if you see one of the hind limbs was black, and whereas the rest of the three rest were white. And people were saying, this is fake. Dolly does not exist. Dolly is a fake. Dolly has been a product of the imagination of the researchers. Well, the reality was a bit more simpler. In reality, this front page was obtained from this previous picture, in which the hindling is not from the lamb, is from the mother, which is from another strain that has the black feet. So therefore, in this case, that was easy. But more complicated was when in 2012, the Nobel in Medicine or Physiology was given to Gordon and Yamanaka for reprogramming cells and for converting somatic cells in pluripotent. of course, they forgot the Scottish, they forgot Ian Wilmot, Keith Campbell, and the Roslin team. They really forgot, or was something wrong with it? Well, let me ask you, who among you know Angelika Schnicke? You might activate your camera. You know Angelika Schnicke? Well, you should know Angelika Schnicke, not only because it can be also a role model and is an extraordinary woman researcher. She's the one that was doing the experiments that led to the birth of Dolly. That was her PhD. What do you expect when you are responsible of a work and you are such successful? Where do you expect to find your name in the paper? You expect to find this paper, the first author of the paper. Yes or no? Where is Angelika? Angelika is in the second author. Why? Because the chair of the department, who had been battling with this project for more than 15 years without success, he realized he's an admirable person. He's a good friend of mine, but he lost his mind for a moment. And he thought, this is going to be a paper that will be heavily cited. I'm going to be first. And he put himself first. And that created a tremendous discussion in Roslin, especially from one of the PhD supervisors of Angelika, which is Keith Campbell. Keith Campbell is the last author of this paper. And Keith Campbell, he really left Roslin because of this. They were discussing, they didn't find a solution. He went up and he never ever talked with Ian Wilmot again. And Keith Campbell was only left with the idea that maybe in the future, whenever this achievement would be recognized with a Nobel Prize, they will have a chance to be part of the Nobel Prize. In 2012, when the Swedish Academy of Sciences, they chose the awardees, they only chose John Gordon, who had done the cloning with frogs in the 60s, and they chose Shinya Yamanaka, who had been finding these reprogramming factors. But they forgot what was happening in between. Well, they didn't forget this. They specifically exclude this because these discussions arising with authors, they went into the court. So there were people who were doing the experiments, who were doing the injections in the embryos, who were not even named as co-authors of the paper, were not even named in the acknowledgement section. And these people, they filed, they were suing this Ian Wilmot and Roslin. So there was a big disaster behind a paper that should have been a very nice prospect in science. This is often not known. And what was not even known is that Keith Campbell, on the weekend that was known the 2012 Nobel Prize, I think he thought it was the last chance to be recognized, to be credited. He probably knew that they will not be named into the prize. This is a speculation. We still don't know this. But on that particularly weekend, so this news were known on Monday, he hung himself. See, he committed suicide. So you see, a paper that could have been a nice history, it's contaminated by someone claiming to be first author and resulting in a controversy that eventually leads to the suicide of one of the main authors. So this is an example of a paper that because of this research misconduct ended up not being recognized with this Nobel Award. By the way, Angelika, she's been progressing and she's one of the professors in the Technical University in Munich. And she's one of the leaders of xenotransplantation with pigs, genetically modified pigs in Europe. And she's been often the chair of many committees. She has now recognized their credit, but at the time, she was not recognized. And this is something you need to take into account. Woo-Suk Wang was claiming to have been able to obtain patient-specific embryonic stem cells from human somatic cell nuclear transfer blastocysts. Please have a look. Please have a look on how many authors these two papers in Science in 2004 and 2005 were published. Well, nothing in these papers was right. They were all invented. So everything that is in these papers was a fraud. It was a fraud that was in 2006 that was retracted, these papers. It took 2 years to find out. And it was found out because some of the eggs that were used in the experiments were kind of donated by PhD students of Woo-Suk Wang in Seoul, in the Seoul University. And he was saying that he will be compensating their students with some money, but he forgot to pay. And some of these students say, you don't pay, well, then we will reveal that you have been committing fraud. So that was the cause of this. And eventually, this person had trustability, Woo-Suk Wang, because he had cloned the dog and he was in jail. He had to pay a fine. But he's now back in business. He has been released from prison. He created this company, Sohn Biotech. If you don't know what to do with $20,000 and you have a cat that is about to die, you might wish to clone your cat, or you might wish to clone your dog, and you pay $20,000 or $30,000 to them, and they will clone your cat and they will clone your dog. So that's the second life of Woo-Suk Wang. Then we have Haruko Obokata. Haruko Obokata, probably some of you know this person. She was a PhD student from Riken. Riken is the most prestigious research institution in Japan. And she had 3 different PhD supervisors, 2 in Japan in different cities and 1 in Boston. And she of course had a talent because she was telling different things about the progress of science to the different supervisors. So she had to maintain 3 lines of lies. So you need to have a very organized mind in order to maintain these lines and not to be caught. Eventually, she managed not to 1, but 2 papers in the same issue of Nature. And she was claiming that you just have to change the pH of the media in order to convert the somatic cells into a pluripotent cells. Well, this was fake, this was fraud. In this case, it was only a couple of months in which it take to realize that that was a fraud and that was not possible. Okay, the consequences were dramatic as well. One of the supervisors of Haruko Obokata in Kobe, at the Center of Developmental Biology in Kobe, the CDB in Kobe, this is Yoshiki Sasai. And Yoshiki Sasai was one of the most advanced developmental biologists in the field of pluripotency. And he could not resist the shame of being associated with this case. And he was hanging himself at the main entrance of the CDB in Kobe. So he also committed suicide. And he left one note for Haruko. Let's make sure, and please make sure that your experiments are right. And the experiments from Haruko, they were not right. So you're not only altering the record, you're not only committing fraud, you also bringing people to commit suicide. Eventually, it was found out that she was mixing up embryonic stems from a different origin, and she was claiming these cells had been appearing from the pre-existing somatic. That was a total fraud, and that was a total discovery. Of course, we had other types of research misconduct, such as with human embryos. And this is He Jiankui from Shenzhen in China, 26 November 2018. He claimed to have been able to produce the first girls, the first human beings with their genome being edited. I already told you in the previous class that those countries that they have signed the Oviedo Convention, they cannot modify the genome of the descendants. Those countries that they did not sign does not mean that they are permitting this. They have, at the time, China, they had recommendation not to do this experiment. Of course, he crossed many lines. He was thinking about removing this receptor, CCR5, because that's the receptor used by HIV. But of course, the receptor is not there to allow the entry of the virus. It's there just because it's a regulator of the immune system. And if you remove it, you will cause a number of problems, such as increased infectivity by other viruses and increased mortality after just a simple influenza virus. So he was presenting this in the summit in Hong Kong in 2018. He was cheating, he was fabricating. This is the falsification of the approval of the ethics committee approving this. So she was falsifying. And when some journalists found out the details of the research, he was evaluating these embryos that had been edited before being implanted in women, and he knew that he had not obtained the modifications he had planned. He had been modifying other genes, so the best he could do was to destroy these embryos. And even knowing this, he continued, and he was transplanting. He was cheating the gynecologists and telling them to implant these embryos, telling them they were normal standard in vitro fertilized embryos, but they were genome-edited embryos. And he was sent to jail, and he was sent to a fine, 3 million yuan, and inhabilitated. And of course, this is something that you cannot do, because there's so many things that happen in the embryos, and this was later on confirmed by a number of researchers. The problem is that there are 3, not 2. At the beginning, there were 2 twins, and there is a 3rd girl. So there are 3 girls with the genome edited, which might suffer the consequences. And this is the result of an experiment that should not have happened. He just has been released from prison. And this is He Jiankui again, with a bit less hair. And now he's claiming that he will be solving the people that are suffering from Duchenne muscular dystrophy. Well, I don't know. This is a toxic person, and this is a person that should not be allowed to go back to any lab anymore. So, and this is, for instance, what's happening with genome editing. Another type of research misconduct is when you try to apply this for improving. This is a biohacker. This is Josiah Sainer, and you can learn about him if you follow this TV series, Natural Selection. He wants to inject himself a guide against the gene myostatin, which is a negative regulator of muscle proliferation, in order to get similar to what is happening in nature. And this is a cow in which the gene spontaneously, the myostatin gene is mutated, and of course, it is hyperproliferating. Of course, this cow, besides having this number of muscles, has many problems and has altered animal welfare. To begin with, these animals cannot be born normally. They have to be born by cesarean, and they are suffering from cardiovascular disease and many other things. So you see that these proposals for applying this onto human beings, they have to be considered carefully. So how are we inspecting? How are we paying attention to what other people are doing? How are we discovering these frauds or these falsifications? Well, there are 2 different tools, one that I trust it more, and one that has to be used if you want to use it carefully. Retraction Watch, as the name indicates, is a list of papers that are being retracted with the corresponding explanation. There is not much to be said. Basically, once the paper you are involved with is retracted, you will be appearing in this web. That's it, period. But here is something else, because through anonymity, an anonymous person can denounce and can complain or can say that researcher in that paper, in that figure, has a manipulated image. And this can be true or cannot be true, but already the damage has already been done. And there is no consequences for the person that has been revealing this fact, especially if the fact is not right. And this is why I'm saying that peer, even though it can be seen as a democracy in science, so everyone can reveal any misconduct, the reality is that can be very easily misused. For instance, these are examples in which the same blood is being used in different papers, published in different journals, representing different proteins. This cannot be a mistake. This is a manipulations done in purpose. Or these are bands that are being reproduced again in different papers. And this is the case. One researcher we had in our country, in which whenever they were finding some manipulations in their recent papers, when they tried to go back, they found misconduct and manipulations during the principal investigation when she was a postdoc and even during the PhD. So basically, this is telling you that the research misconduct is a pathology. Some people, they want to apply this, wrongly, of course, but then you need to have a talent to do this, and you think that you are able to cheat everyone. You try it, and sometimes you try it and it works, and you say, well, I will be raising my bet, and I will try it again, and you do it again, and you do it again and again and again, until you lose control, and eventually you're found out, you are caught. And when you are caught, people is just going backwards in your publication registry, and they will be revealing and they will be finding everything. What are the standard reactions and responses that you're getting from people committing research misconduct? To begin with, to my knowledge, I have done nothing wrong. All these accusations are false. So this is very classical. There is an easy explanation for each mistake, and that could be some inadvertent mistakes. I could easily replace the figure with correct data, but what a curiosity, I have lost the original raw data. Or, this is very often the case, my computer just has been stolen and I had the data there, and I'm sorry, I cannot use the raw data because they were in the computer and the computer is no longer with me. Or for instance, something that you would not be able to say from now on, I did not receive education on ethics and research integrity. And this was the defense that was acted by Haruko Bokata in Japan. And she was right. RIKEN did not provide classes on ethics and research integration to RIKEN students. And because of Haruko Bokata, and because the judge in court, she was giving her the right, so she was accepting this defense. She did not receive, she did not know she could not reuse figures. She did not know she could not improve figures for papers, even though you might think this is common sense. You might think this is a bit naive, but the reality is that the people in the RIKEN institutes in Japan did not receive. So because of Haruko case, nowadays, everyone that is entering any RIKEN institute in Japan, before touching any pipette, it is mandatory that they follow these classes on ethics and research integrity. We have cases in which there is a reuse of control names. Here we have a case of Nature Genetics paper in which these two figures that are framed in red, they seem to be different, but in reality it's the same figure that has been turned around. And this is my favorite. This is a champion of reusing of bands, and so the same bands have been reused. You follow the code, the color code have been reused for different products, for different things. So basically, this is happening. Other similar issues that have happened recently that should put you aware that you can be caught even by some very minor details that you are not in control. These are two Swedish researchers that they published a science paper in 2016 that happened to be retracted in 17 because this data was not, they were fabricated. So there was a fraud. And they were investigating the interference on larval fish ecology by microplastics. They were doing this in the Baltic Sea, and they were doing this, and this is the retraction, and this is the two researchers, the person supposedly doing the experiments and her supervisor. And basically what happens is that they were caught because of Facebook, because they went to Gotland. This is an island in the Baltic Sea where they claim to have performed the experiments. What they brought, this is up to the left, they brought in the science paper, they have been collecting data over a 3-week period, but they have been taking pictures of themselves in Gotland, and in Facebook, they document the arrival in the island on the 4th of May, and they document leaving the island on the 15th of May. So this is 11 days, not a 3-week period. So they were caught because of this in Facebook. No? Other very popular retractions, still we're paying the consequences, is this Wakefield paper. This Wakefield paper that was linking the vaccines with autism. This is very often being used by anti-vaccine groups. Even though this is unjustified, false, and imprudent, even though this was retracted, this is still being used. And the consequences is that the number of measles cases, because families, they're not vaccinated against measles, are increasing. This is in the US, but this is also happening in Europe. So these are the unwanted consequences. Another type of research misconduct that is, and I'm using an example of our institution, is the ghost authorship. So some author that nobody knows, but is providing data, because all with what we know from this person is that he's sending the results that, surprisingly enough, these results, they always fit with the hypothesis, and this person is sending always messages from a Gmail account. So this person that is pointed with a red arrow never existed. This was a ghost author. Nobody dared to double check the list of personnel in the institute and to find out that this person wasn't there. And actually, this was an existing person. This was a university mate of one of the authors committing this research misconduct. So he was using the personality of his old friend to be claiming that this person was providing some results that were supporting the case in this research paper. Of course, you see that you are a co-author on some of these studies with multi-authors papers. Well, you need to know, and this is often neglected, that you are co-responsible. So you might say, I have nothing to do with this figure. Figure number 2, I haven't done. Figure number 2 is the one that is fabricated. But you are a co-author in a paper that includes figure number 2. So you are co-responsible. If you are not trusting this, what you need to do, you don't have to be co-author of this paper. You have to remove yourself from the author. But if you stay as a co-author of that paper, you are co-responsible because you are validating with your name, you are validating not only your experiments, but the experiments of everyone. So what can we do to prevent, or how can we explain the misconduct? Well, clearly the pressure to publish and to succeed, pressure to publish in high impact journals, the willingness to succeed or to be recognized, and particularly to access to top grants. In Spain, an average project at the most for 3 years will be around 200,000 euros. But if you go for an ERC, in 2-3 years, you will get 2 million euros, which is a factor of 10. So definitely the temptation to do whatever it takes, whatever it needs to be done for accessing to ERC with your longer list of Nature papers is very high. The temptation is very high. And this is what probably lies behind some of this bad criminal behavior. How to prevent? Well, guidelines. what you have to do, what we're doing now, we are studying research integrity. We need to know the existence of the codes. We need to read these codes. We need to use checklists. We need to use these lab books. You need to document everything you're doing with a date on a book that you cannot take pages apart. So you have to use proper lab notebooks, and you have to follow these checklists before you report any publication. Write everything in your lab protocol book. You know this. This is important. If you suspect that there is any research misconduct being applied, you need to get in touch with the research integrity officer, if there is, or the corresponding ethics committee, if there is, or the responsible person in the institution, if there are none of these first ones. There is very important the culture of mentoring and supervision, because we as supervisors, we are responsible of your education. So if you're committing fraud, you are not only the guilty, we are also the guilty because we have failed supervising or mentoring, or we have failed detecting your fraud or detecting your manipulation. And this is something that you need to be developing during this education progress. And basically, mentors have a special responsibility to help the new researchers, the younger researchers, to realize the relevance of the ethical dimensions of research, particularly because we as heads of the laboratories have the overall responsibility on the quality and the integrity of everything that is happening on research in our lab. So this is why we have a particular interest that our trainees, our PhD students, understand the responsible. So we need to understand that young scientists, as you are, at the early phases of their career, they might not know that some practices are wrong, and worse, they might not know or might not be about the consequences of committing this research misconduct. And it is our responsibility to let them know. So this is, for instance, what happens to Olivier Bonnier, that he claimed that he was trained to do things in that way. And at that time, he did not know that they were wrong. So basically, most of the problems associated with Olivier is because he had in mind what the result they should be. He did the experiment, but the experiment was not as nice as he would have predicted. And because it was not nice, he was changing the results so that they will fit better. So he said, I was helping the reader to understand the idea. Well, you were not helping, you were manipulating the results, because some of the results probably they were not as clear as you might think they were. But you see, here's an example. And the same was happening with Haruko. So we have half an hour left, and let me drink a bit of water. And now I have the last part of this research integrity, which is to do with conflicts of interest and the impact of the research integrity in our careers. Well, conflicts of interest. Everyone has conflicts of interest. Of course we do. Because if we don't, then basically it's because we know nothing about it. Of course, when we are evaluating a paper from a colleague, of course we have a conflict of interest. And of course we need to be doing all what we can and beyond to be objectively. And we are not favoring or we are not punishing this person just because it's a friend or because it's a competitor. No? Conflicts are situations where financial, personal, other considerations have the potential to compromise judgment. I mean, I'm evaluating a relative, my daughter, or I'm evaluating my son. Of course, I will be biased. I'm evaluating my colleague down the corridor that I know. Well, I have a conflict of interest. I'm evaluating someone with which I have published a couple of papers. Well, I have a conflict of interest. Then institutions, they put some regulations, and these are variable. For instance, in order not to show conflicts of interest, you should not have published a significant amount of papers in the last 5 papers. What is a significant amount of papers? Well, this is not said. This is not established. Let's say that Vicente and I, we have published 1 paper in the last 5 years, 1 paper in common. Do we have a conflict of interest? Well, I would say with 1 paper, probably not, but we need to declare that we have this paper in common because the committee, they should know. But instead of 1, we have published 15 papers in common. Therefore, we have an obvious conflict of interest and we should not be able to evaluate one each other. They influence behavior and they can be acceptable if handled properly. And what does it mean handled properly? It means that even if you think it is not relevant, you have to declare them all. Always declare, I knew this person, I was in contact with this person, I had a project 10 years ago with this person, I had a paper with this person, etc. You just indicate it because then everyone else in the committee, they will know that your opinions might be biased because of this conflict of interest. And depending on how strong is this conflict of interest, you might be invited to shut up and to don't say anything during this committee, or you might be invited to leave the room because you are not supposed to be listening what we'll be discussing about this person. No? This is particularly damaging when you're talking about the research that is being funded by companies. There's nothing wrong about a company funding research. What is wrong is that the results of this research to be reported more favorable so to benefit the company that has been paying for that particularly research project. For instance, 37% of the review articles on tobacco smoking, they said that passive smoking is not harmful, but 73 of these were scientists with tobacco industry affiliations. So what a surprise. So basically you are paid by the tobacco industry and your findings are that passive smokers, they shouldn't be worried. Well, we know now that everyone should be worried that smoking is dangerous, not only for those smoking, but also those that are sitting next. This is the case of this other researcher, Nashan Chong, and he was claiming to have a product that was doing well in a COVID-19 clinical trial. And he was telling about this, but he forgot to say that he was owning the company that was producing this own product. So obviously, this is a case in which you need to know the others. I mean, there is nothing wrong that you speak favorably about your product, but you need to know that you have a tie with this product. I'm producing this product and I'm saying that my product is the best in the world. Well, if I'm listening to this, and if I know that you are the producer of the product, I might put into brackets your quotation that this is the best product in the world, because maybe this is not true, all right? But because I know that you are the owner. But if I don't know that you are the owner of the company, I might be tempted to believe that this is a very good product. No? So the studies sponsored by industry are more likely to draw conclusions that are favorable to the industry, and we can harm humans, animals, and the environment, and we might endanger the public trust. A chapter in conflicts of interest is when we're acting as a referees, or when we are reviewing journals, or when we are reviewing awards or grants in a selection committee. No? This is an activity that is now occupying millions of papers and hundreds of thousands of people. No? So if a reviewer works at the same institution, it is a close collaborator, it is a close personal relationship, we have a financial interest in common, we have a conflict of interest. And there are new conflicts of interest, for instance, those that are documented here in the Journal of Infometrics, which is a journal that deals with these aspects. referring to what happened with some +1 papers. Some editors, they use their influence to force the authors to let them say, so please add these citations in your paper, and if you add this citation, your paper will be accepted. And these citations were from papers from the same editor. So therefore, the editor was increasing the number of citations and was increasing this H index, and it was increasing absolutely artificially the prestige. So this is another conflict of interest. Where do we have conflict of interest? For instance, in the European Medicine Agency, whenever we have to decide whether we approve or not a medicament that has been produced by a given pharmaceutical company, well, the committee has to be very clear about what are the ties of all the members of the committee. If I'm paid by the company that is expecting this drug to be approved, then I will probably battle for the approval of this product, but I will have an enormous conflict of interest. We too, in the CSIC, we have a manual of conflicts of interest, and I'm also inviting you to browse because it's also in English. You might also look in your institution whether you have manuals of conflict of interest. These are less popular, and these are less frequently found in the institutions. Codes of good laboratory practice are often present, but manuals of conflicts of interest, they are normally not present. So in summary, regarding conflicts of interest, what to do? Declare any potential conflict, even if it's very minor, even if it's minute, declare. I had once a discussion, a public discussion with this person in a Congress. This was a public discussion, and everyone knows that I'm opposing to whatever this person say. Well, you need to say this because your judgment might be affected by this. And then you need to seek advice because the committee will tell you whether you can remain in the committee silent or whether you have to leave the room. Okay, we have 20 minutes left, and now we can discuss about how common is this research misconduct. Well, in 2005, Nature was publishing a paper that was very moving. It was very controversial because basically the estimation was that 1 every 3 scientists were confessing to having sinned, to having committed research misconduct. 1 every 3rd. So this is 33% of researchers. So it means that research misconduct, if we think that these results are representative of what is happening in the research community, this is more frequent than expected. What are the data we have on this? Well, Retraction Watch has a database of now it's more than 20,000 articles that have been retracted. And in 2018, they were revealing and they were publishing the contents of this database. And here we have some misconceptions and maybe some preconceptual ideas that we have to reevaluate, because it's very often to say, where is the burden of the misconduct? And as you can see, so most of the cases are fraud and or errors, but most of the cases have to do with fraud or with fabrication. Well, you might say, what are the highest retracted authors? And because we might be speculating, well, they will be Chinese, because it is assumed that China is where the authors should be more often causing this research misconduct. Well, this is just not true. Look at these top 10 retracted authors. The first one is the Japanese, the most prestigious country, the one that is considered to have more cleanest research. We have Fuji with 169 papers at the time. The second one, Germany, Joachim Bolt, almost 100 papers being retracted. Well, maybe China is in the 3rd place. No, 3rd place, Netherlands, Staple, 58 papers retracted. Fourth place, Taiwan, Chen Yuan Pete Chen, 43. Fifth place, another Japanese, Sato, 43. And only in the sixth place is when we find the first Chinese researcher. So you see, we might be tempted to believe things that they are not accompanied by data. And of course, we have people from United States, from Korea, and etc. The same with countries. We might say, China should be the first. Well, China is not the first. Retraction rate per 10,000 papers, the first one, number one is Iran. Then is Romania. Then is Singapore. How many times we put in Singapore as an example of a country, as an example of research integrity, as an example? Well, you see, it's number 3 in the countries that are committing research misconduct. Then it comes India, Malaysia, South Korea, and then only then China, Turkey, South Africa, and even Netherlands. We have Netherlands there as well. So we have a number of preconceptions that are not necessarily true. And this is actually educative, just to say that research integrity is more common and more often present than what people expects, what people assumes. How common is in our institutions? I don't want to use any other example. I'm going to use my example, my institution. Well, this is what I was evaluating while I was chair of this institution in the last 2 years. And if you see, in total, we have 155 cases that we evaluated. Are they many? Are they? It's a very high number, it's a low number. Well, I think it's a respectable number because we have more than 13,000 researchers, and this represents about 1.2%. And I think similar to what happens, this is the 1% death consequences of the coronavirus infection, and so many hundreds of thousands of people have died. So 1% of this, it's already a lot. And you see that the number of cases per year might vary. And curiously enough, in the first year of the pandemic, this is where we had the lowest number of cases. But later on, the next year, we had more even cases. So on average, we are dealing with 11, 12 cases. So it's almost a case per month that we are dealing. And this includes fabrications, plagiarism, this includes copying, this includes cheating, this includes all the different aspects of this research misconduct. The best study that has been evaluating how common is research misconduct is this study by Fanelli and from Edinburgh. She was preparing an analysis of 21 studies evaluating research misconduct. So this is, I would say, is a very good representativity of what is going on in reality. And what I will be presenting next are the conclusions of this meta-analysis. Normally, what I do, in this case, I will not do because you are so few, there's only 10 of you. But when I'm teaching about research integrity for 50, 60, 100 people, 100 students, I'm running internet test live, and I'm asking the same questions that Fanelli was asking in this analysis. And surprise, surprise, the percentages and the numbers that I'm getting, no matter which institution I'm visiting, are very similar to the conclusions that were published by Fanelli in 2009. So what are these numbers? 72%, so 3 out of 4 people, they say they have observed questionable research practices by colleagues at least once. What is a questionable research practices? Well, removing 1 point of a series so that the resulting is statistically significant. This is a questionable research practice. 3 out of 4 have observed things going wrong in others. 14% of people have observed fabricating or directly falsifying this data by colleagues at least once. 14%. 40% is almost 1 every 7 or 1 every 7 researchers. This is a lot. 34%, 1 every 3, they admit themselves to be practicing questionable research experiments at least once. So people is admitting that at least once was manipulating an image or was manipulating some data. And last but not least, and this is the number I want you to take home, 2% admitted fabrication and falsification. So these 4 numbers, whenever I do this experiment live with people, anonymous, so I don't know who is responding what. This is using one of these applications that you can collect answers. I'm getting systematically the same values. I'm getting very often the first question, rather limited the second questions, again, very, very popular the third question, and roughly between 1 and 3%, so 2%, I would say is a good average. What does it mean, 2%? Well, what it means, I don't know if this 2% we have to take it immediately mathematically. What it says is that this is not a 0%. This is what it says. What it says is that we, the researchers, we are not different from any other group in the society. We are not better, we are not worse, but we are human beings. And because we are human beings, we have temptations. And because we have temptations, we might be tempted, a limited fraction of us, to take a shortcut. And this is what you see here. There is also a shortcut when a taxi driver is charging you more for a run, for a drive, than he should be charging you. This is also research integrity. This is done by all taxi drivers. No, it's only a limited fraction of them, but those that are causing the problem are really making a lot of noise because people speak about this. I've been charged 50 euros for a drive that should be less than 15 euros, let's say, something like this. Well, in research, we have the same situation. We have an institute with 500 researchers. 2% will be 10 people. Do we have really 10 people committing research misconduct? Well, I don't know. Maybe we have 6, maybe we have 15. But what I do know is that we do not have 0 people. So this is the last thought I wanted to leave it with you. Basically, my final recommendation, try to be a good person, a good citizen, and you will be a good researcher as well. So you can find myself under this email address. You can find what we do in this web page. And now we have like 9 minutes for any questions or comments that you may wish to have. So thank you. Questions?
Okay, thank you, Luis. So really, really nice presentations. And
at least once they should hear about this.
Yeah. No, it's impressive. And I mean, both parts. So the first one touching very sensitive points in ethics, use of animals, regulations on that is actually particularly important for us, no? The gene editing at the European level or at the worldwide level, no? What is going to be the regulations and so on we can do.
Let me ask the students, what do you think about this final conclusion? About 2% of us were committing research misconduct. Did you notice? Did you expect this? Anyone?
I can talk a little bit about it. So I didn't know. I had no idea about the percentage, the usual percentage that is always there. But I mean, I never thought it was 0. So I'm not surprised it's not 0. I just didn't know how much it would be. I had no idea.
This is the correct interpretation of this number. Don't pay attention to the number 2, but pay attention, it is not 0. So, which means that we are not different from any other group in the society. And basically, because we know this, because now you know this, because now you know the consequences, and Did anyone know about the consequences of the Dolly paper, the Nature paper? So you see, you didn't know. This is one of the most famous papers in biology, the cloning of Dolly. And it was a disaster at the personal level. Someone was occupying the first position of the paper abnormally, and someone committed suicide because the paper was not included in the Nobel Prize later on, years thereafter. So you see the consequences. So even this Japanese girl that was committing fraud and was leading to the suicide of one of her 3 supervisors. And I know the other Japanese supervisor, and I know him personally. And I've been talking to him, and he basically admits, I had no idea, and I have to believe this. I have no idea what this person was doing. I was just judging whatever she was presenting to me, and all what she was presenting was according to what I would have expected. Of course, if someone is presenting you a blood or a PCR, and the PCR is showing some bands of the appropriate size, you have to trust your student. There's no way you can say, I don't trust you, so please do the experiment again in front of me so that I know that each lane corresponds to each plan or to each individual. You have to trust, because without trust, there is no research, but because we're trusting, then sometimes we might be facing these consequences. Lab books, write everything, put a date, okay? Depending in which country you are, you have to write in pencil. And why pencil? Because if the lab book gets covered by water, it will not be blurred. If you're using ink and all of a sudden you are disposing alcohol, then everything will disappear. Pencil will not. Pencil you can erase, of course. There are always pros and cons. But definitely lab books. And when you're writing a lab book, you're not writing only for yourself, for whenever you have to collect everything and prepare a PhD manuscript or a PhD paper. You are collecting this for anyone coming later on. So write phrases. So it's okay if something is going wrong and you put one day, shit, or fuck, or this is something went wrong. But please explain, what is this blood? What is this gel? What is this microscopic picture? This is this, I was expecting to find this, but instead I found this other thing. And this is now kept and can be used by someone coming after you. And the multidisciplinary articles, this is something that people is not aware of this. You are co-author of a multidisciplinary article. This article is retracted. You are co-responsible. Okay? You might claim, I have nothing to do with the experiment that went wrong. You are co-author. You said you bought this system. Shannon.
Yeah, I have a question. Maybe it's more for Vicente, but it hasn't really been discussed with us, so maybe it's a good time to discuss it. Obviously, we're in a European project which has industry partners in our project. And when you brought up conflict of interest, I was thinking about it, how at a later stage when we want to be publishing the science and people see that we have people working in the priming industry that are in our projects, how that works.
Well, let me say something and then Vicente. It's nothing wrong to have a company part of your project. Actually, it's better because you're showing that what you're doing has some implication in the society. You are translating your research results into a company. The only thing is that you have to declare So what it will not be nice is that there will be a company supporting or participating in your research, this company being part of a study, and this study not being recognized that this company has been part of it. Or if there is a product, there is a seed, there is a variety of a plant that is the product of your research, and this is result of a given company, and you are not telling that the company has been providing some funds, some money for you to do the experiments. There was a very popular case in cancer research. There was a very known, actually it was a Spanish oncologist, and he was very prestigious. He was even appointed the chief of the Sloan Kettering Institute in New York. And he was obtaining funds from Roche, from the company Roche. And Roche was producing some anti-cancer drugs. And he was publishing how good these drugs were functioning in cancer. And because he was so prestigious, people were following and using these drugs. He forgot to mention that he was receiving money from this company. And because of that, he had to resign. He was fired from the Sloan Kettering, and he had to resign his commitment with many societies. He was expelled from any other society because he was committing a research misconduct. There is nothing wrong to have money, to receive money, but you need to declare it.
I would like to add that in the case of Episeedlink or all kind of Marie Curie actions, because we are regulated by the European Union, we have to sign a consortium agreement. So it means that we have already agreed with the companies what can be published or what needs to be protected by patents or any kind of protective measures, no? And everything is in a document, but also there is in the meanwhile, during the progress of the project, we are elaborating a data management plan. So that also is updating all that kind of ways to publish or to protect all the results in each of individual research projects. So everything has been agreed. Actually, the consortium agreement took more than 1 year to be signed by all partners, discussing all these kind of protective measures, how it's going to be published or not. And also at the level of the grant agreement with the European Union, we have to indicate for each of the activities of the action of the project, what is the level of protection for each of them? So what is going to be public or what is going to be confidential? So for everything, training, research, so any kind of outreach activities, publications, books, everything. So it's already indicated. So, but anyway, it can be updated during the project.
Yeah, I think it's just nice for us to know as well that all of these things are done. We don't have to worry about them. We're just not, I think, discussed about it yet.
It's good to know. It's good to know. You're right, Susan. Shannon, sorry. I think we have time, but still, if anyone has a question.
I have one, if I can. So it's about in the first part, you were talking about all the animals, no, that you have to reduce the as maximum, no, the number of animals that are used, not in the sample, but in general, no, or to. But what happens to animals that are after they are used for experimental purposes? Are there spared? So, for example, in the case of macaques or something like that, if they have been treated with any chemical or with the
placebo. This is an important question, and the directive takes care of this question. Basically, we are obliged to consider reusing animals. In case the animal welfare alteration that has been committed to that particular individual allows, after some recovery time, to be treated again. So this is applied for dogs, for cows, for horses, for lambs. When we are using all these species in research, for instance, you are evaluating the insulin response in a sheep model, and you are feeding this animal with a given diet, and you are investigating the hormone levels. Well, after the experiment has been completed and you have taken the blood samples, well, the animal is still alive. Animal is still there and can be recovered for some time and can be reused in another experiment instead of using another sheep, which is different. Then this is something that we need to take care. But for instance, for dogs and for primates and for macaques, in most of the cases, in the few cases in which dogs and macaques are being used, normally the research goes to the end. It means what you do to an animal eventually results in a pathology experiment at the end. So basically you have to sacrifice the animal and you need to prepare all the organs and et cetera, et cetera. So this is more often with livestock, with farming animals. And for instance, with mice, this is not applying. So basically mice are discarded and we are not reusing these animals.
Okay. Anyone?
All right, then I hope you enjoyed these 4 hours, and I will be providing these slides. I will be clearing, because I had a couple of repetitions. I will be clearing this and I will be providing this to Vicente, and Vicente will distribute this among you. And I wish you all the success and plenty of success in your future careers.
Thank you very much.
Thanks.
Bye-bye.
Thank you.
Thank you, Luis.
Thank you.
Bye-bye.
Thanks. Bye.
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 Thank you. Bye.
Okay. Anna, are you still around?